It's like,
Here
there
we
it
are.
is. There it is.
There it is.
Here we are. Phil. We are. I feel like we're vastly outnumbered in terms of IQ here right now.
I would concur. I would concur graciously. So
This is episode 102 of Snakes and Stogies, brought to you by Puget Sound Pythons. They're right here.
my box is my square
Everyone
is different.
struggles. And this episode is going to be a good one. As I'm sure a majority of us know, there's been a lot of stuff going on in terms of like morph market and a testing policy, and then a reversal of that policy. And so now we have the best of the best in terms of like the greatest minds of herpetoculture, and then when it comes to pathology and stuff like that, and
The cream of the crop.
That's right. I mean, over here to our yep, my right, your left, other way, buddy.
That way.
Yeah, we're already making this look really whack.
Yeah, that's all right.
This is Dr. Zac Loughman, who we've had on THP at least twice. Our buddy Dr. Travis Wyman right below me, and Phil here. Yep. And then Steve Tillis, underneath of Doc Loughman and Phil. We were going to have Dr. Julander with us. He had to bow out last minute, so that's okay. We have plenty of good people to give us their 2 cents on the matter.
I messaged the good Dr. Julander sometime a little while ago, and he said he will be listening, and he will be here in spirit.
Sweet.
That he is.
Good to have him in some form or other.
Of course,
Christian.
100%. Christian. So yes, 102, gentlemen. 102. Yes.
What are you smoking on? Anything? We'll get cigars. I'm trying to we're gonna kind of breeze through all the intro stuff because we got a lot to get to.
We got a lot to get to. I actually had a beautiful limited edition VSG that it's in the house somewhere that somebody gave me, but I'm still getting over the worst sinus nonsense I've had.
Power through it.
Worse than COVID, like just gross.
Smoke through the pain, Phil.
Well, that's when I am smoking through the pain. God bless. But my my palate is tainted temporarily, so I felt I should not try to smoke a cigar and not like it. You know what I mean?
I am officially taking online courses to be a certified retail tobacconist, and then I'm going to be a certified master tobacconist at some point too. So
excellent! Congratulations in advance.
It's like a DeVry version of a certification, so let's not make it sound fancier than it really is. I mean, I'm no Doc Wyman by any means, but
you'll still be a sommelier of tobacco.
That's exactly what I was about to say.
Yeah,
I mean that's pretty much what it is. But I'm smoking a Oliva G Maduro, and then once this is done, because I feel like this is going to be a 2-stick show, La Imperiosa from Crown Heads.
Nice,
it's a phenomenal smoke, especially in this Toro size.
Love that.
And I made them both long because I feel like
good.
I'm going to need them. And I got my handy dandy can of heart disease right here next to me as well. So.
Delicious.
We're off to a great start.
Is any of the academics in this group partaking in any adult beverages and or tobacco?
Nope.
Yeah, I might grab a beer halfway through, but yeah, we'll see how it goes.
Fair enough. So, Doc Wyman, you want to kick this shindig?
All right. Yeah. So this was kind of motivated by the eruption, as it were, that hit last week after Morph Market had that policy announcement and then walk back. And I felt this was kind of necessary, not directly because of that, but just indirectly because of so much of the chatter going around.
It sort of kicked things up a little bit. It stirred things up.
It kicked things up, and it kicked up that there's still so much misinformation going on about there. And a lot of it was driven towards Nido, but I think there's just a lot of misinformation in general about disease in reptiles. And so it seemed to me that since the hobby, the hornet's nest has been kicked and everybody's kind of attention is up. Now would be a real good time to try and discuss this again, get more information out there for people so that we can try and knock down some of this misinformation, kill some of these rumors, and try to get people thinking more about this in a proper and intelligent manner than just this reactionary fear factor.
Sort of knee jerk.
Yeah. The kicking and screaming and the hair on fire, which
Yeah.
that doesn't help anybody on, regardless of which side of the fence you sit on.
So should we give like the Cliff Notes version of the policy now, or should we handle that sort of later?
We can give it now, or we can hit it later. I mean,
because I'm still a little fuzzy on it. I kind of understood the gist of it, but
okay. Well, the Cliff Notes version is basically that John released a policy that basically said, a lot of people have this health guarantee on their animals, but it's kind of like, the default one for Morph Market is like 4 hours. And most people have, you have to let me know within 2 hours, 6 hours the next day type of thing. And John recognized that there has been an uptick in people who get their animals tested for these viruses, but the turnaround on that is not within 2 hours or 4 hours or anything like that. Right. So he was writing in this policy that basically said, if somebody gets, wants to get one of these tests done, since it takes greater than 4 hours to get the results back, as long as they get video or picture proof that they're taking the tests and sending them in within 2 hours of receiving the animal, then if, it takes 24, 48, a week, 2 weeks, depending on what lab it ends up going to and coming back, if the animal tests positive then, and there's a dispute between the buyer and the seller, if the seller doesn't have something explicitly written in their own terms of service, then Morph Market will side with the buyer.
Okay.
So personally, I don't feel that's unreasonable. And I want to clear something. It's not widely known by a lot of people, but I am sort of technically classed as Morph Market staff. Anything that I'm talking about tonight, I'm talking about from my own personal perspective. I am not speaking as a representative of Morph Market. I think John has made everything clear there, and John is ultimately Morph Market, so I'm not speaking for him. This is all me speaking as Dr. Travis personally, not Dr. Travis Morph Market staff, and my position on staff has nothing to do with making policies and things. I help moderate the forums. I will help sometimes if there are, questions about morphs that are put up in the market itself that don't look right, but I'm not making policies or things like that. So don't go gunning at John saying, well, Travis said this as a representative of Morph Market, because I'm not saying that. I'm saying as me personally, I think that was a legitimate policy to put in place, because if people are testing and they do get something that comes back hot, obviously it's going to take more than that 4 hours.
Right.
And I don't think that it's unreasonable that if a seller has said, you only have 4 hours, that's kind of, impossible to give a viral test or a bacterial test or a crypto test or something like that to come back in that time. But then everybody blew up over it, and there was a lot of, you can't force me to test my animals, you can't force the hobby to test animals. That's not what this policy was saying. It wasn't saying you had to test your animals. It wasn't saying you had to register as being a seller that tests their animals. It was basically just saying, we're putting this new policy in that says, if somebody tests their animal, it comes back hot, unless you specifically said we don't accept disease tests.
Right, that was the opt-out.
Right. You just have, and if you don't want to do that, you could change your own terms of service to say, we don't accept testing. And, then it's like, well, you're doing that to just blackball, you're not blackballing people. It's not what it's for. And,
Right.
It blew up. And like I said, so much of the blow up was to me very obvious that it was based on a lot more just misinformation on, like I said, on both sides. You've got both sides pointing fingers at each other and screaming and yelling at each other. And that doesn't help the hobby as a whole. So I felt getting people in the know. I know that Zac has done a ton with crypto because he's had to deal with it up in his collection. Steve is literally studying this virus. And so, who to go to better than somebody who's actually, on the cutting edge and the breaking wave of it. I don't work specifically on snake diseases, but I work on how to test samples for, diseases and things, very cryptic, hard to find things. I work a lot of sequencing. I work a lot of PCR. I had wanted to get Justin in for the same thing. He's a virologist. He knows about, Nido in general. He knows a lot about viruses in specific. So unfortunately, we don't have him here, but, we got his input some on the stuff we're going to talk about. So this, I hope, will help bring tempers down some on both sides and maybe get people to see things in a little bit more clear light coming out the other end.
100%.
Yeah. So I know like Nido and crypto are kind of the two big ones that are sort of the bulk of the conversation currently, but I know there was other things that you wanted to talk about that aren't as commonly seen or just aren't as prevalent.
Yeah. Well, I don't know that they're not as prevalent. They're just not as frequently talked about. So, the major things that we see are crypto, and basically anybody who works with colubrids knows about crypto. Nido has become a big thing. It's primarily pythons. You have the arenavirus IBD, inclusion body disease, in boas, which also is a problem for pythons. In fact, it tends to be a bigger problem for pythons because boas seem to be just kind of naturally resistant or resilient to it. But if a python gets it, almost inevitably it wipes the python down. But you have other diseases that don't get a lot of talk about because they just don't make a big splash or people are unaware of them. Adenovirus, rheovirus, iridovirus, paramyxovirus, they're out there and they're not insignificant in some cases. So people don't talk about them, but they're there. And it's very possible and probable that some of these situations that people are seeing and hearing and finding have nothing to do with Nido or crypto or IBD, but are, one of these other ones, and people don't know about it.
Yeah, I would definitely say that is pretty accurate. There's a ton of pathogens out there that just aren't being tested for on any sort of broad level. And, if there's one thing that working reptile diagnostic testing has told me is that if no one is testing for it, it is probably safer to assume that it is far more prevalent than people realize.
Right.
I'm sure the number of samples and tests and stuff you see, the amount of positive for anything is probably a little terrifying.
It can be, yeah.
Realizing how big that iceberg is below the surface,
It is and it isn't, especially when you're talking about serpentovirus, because there are so many asymptomatic positives. That's where it does get a little bit more tricky, is trying to determine when a virus is significant to the, either the individual animal or more importantly, the colony as a whole.
Right. And then, correct me if I'm wrong, but there's over 80, right?
Yeah, something like, it's been a long time. It's been like about 6 months since I've done reorganized the database I have for all the genomes. But I think last time I did a tally, it was like 200 something unique genomes. Within that, I think it was 80 had 5% or more divergence from each other. So there's a ton of genetic variation out there in these viruses.
and that genetic variation, like Steve implied, can have both big and small impacts on symptomology, disease causing potential. And we can dig into that a little deeper now or later. I mean, whichever way we want to go. If we want to talk about some of the lesser known ones first, just to knock them out of the way, or we can just jump right into the serpentonitoclade and hit crypto as well. I mean, whatever we want to do.
I think before anything, it's probably worth kind of just recontextualizing what it is that we're actually doing. It's not like we're taking all of these animals originally come from some sort of wild origin. It's not like we're passing them through an autoclave before we set them in a tub, and you couldn't. So these are not sterile things. A lot of what I do is tissue culture, and when we have an animal that dies and we collect tissue and try to make a cell line from it, there's frequently bacteria or viruses just latent in the cell culture itself from the actual animal that might have been in X number of generations in captivity. The line of pathogen doesn't stop the second we put it in a box.
Yes.
So yeah, I think it is important to recontextualize that they don't just pop out of nowhere. All this stuff has some source.
Yeah, it all has origins, and I think that's part of what leads to some of this misinformation is people think, well, I practice good husbandry, and so my snakes are clean because they've all been captive bred, and that doesn't necessarily hold it up. I mean, dogs have been captive bred for millennia, and your dog still gets sick sometimes. Same thing with cats, horses, cow. Basically any domestic animal that you have, you still have diseases that all of them suffer. It's easy to point at somebody who, picks up import animals and be like, well, it makes perfect sense that they've got problems in there. They're bringing in wild animals. But even people who have completely closed collections, those things are still passing stuff around between them because nothing is, like Steve said, nothing is sterile here.
Right.
In the way that we keep them too, the density of animals per square foot for some of these collections, you just literally, it's like the golden corral for viruses, man.
Yeah.
They're just ready to walk right up to that buffet and start
sneezing all over everything.
Mm-hmm.
Yeah. Between the biosecurity or lack thereof around reptile expos and just the general standard, general standard and general density, if you were to design a system to create a pretty virulent pathogen, we've done a very good job of it.
Absolutely.
Well, I guess that leads me to my first real question, and this is extremely rhetorical, but in the past, go back 15, 20, 30 years, whatever it was, when reptile shows were first booming and, people had tables full of animals and reptile pet shops started popping up here and there. Was it that the health issues that we're seeing today in a much more prevalent light, was it that they hadn't mutated or evolved to be the point that they are now? Or was it just, we just chalked up to, oh, it's an import, it dies?
It's a result of complete ignorance.
Right.
Crypto, I can't speak to the viruses. I can definitely speak to crypto. And for the record, I'm saying that I am not an expert on crypto. I just had to deal with it. And since I'm a academic nerd, that meant giving up 10 days of my life and reading every single thing I could possibly get my hands on and mapping it all out. And then talking to people that were the experts, the guys that were writing and the women that were writing those papers. So I've kind of have this expertise, if you will, that's totally achievable for everybody listening to this thing if you just are willing to put in the time and learn and read, because that's where, and then obviously I had to apply to the collection. But these animals, when they're brought in from the wild, one way to look at it is, we all know the horror stories of importation and how you go and there might be, for every tokay gecko that makes it, there's 200 or 300 that die, or maybe 2,000 or 3,000 that die. So in a weird way, from a biological point of view, you could kind of look at it as the animals that are most fit are the ones that are actually surviving to the table. And so when you're just working with those wild animals that we haven't taken their genome that's like this big and then whittled it down to this big, they're stronger, but what that really means is that they're able to hide the disease better. So, they start eating, blah, blah, blah. They appear to be great, but they're not. And one of the things like with crypto, the number of asymptomatic snakes in America right now is probably, I would go so far as to say it could be as high as 40 to 60% of colubrids have it. Easy. And so the question then becomes like, well, what are we actually seeing? And I know that I've heard Steve talk about this with serpentivirus too. And that it's very naive and arrogant, in my opinion, to assume that you can beat this and you don't have it if you have a collection of 50 plus snakes. It's just going to be there. And that's basic biology.
Yes.
So the fact that, Morph Market put this clause into effect, being a biologist and trying to maintain that standard of cleanliness or whatever you want to call it, disease-free, I don't see the problem.
I would agree. Yeah, a lot of it does come down to what the goal of managing any sort of pathogen in a colony is, because if the line you want to draw is absolutely nothing is getting in, that might not be a realistic bar to try and upkeep. There's limitations on testing. Nothing is perfect. Nothing is watertight. So, really, I think the proper mindset to go into it would be to assume that this stuff is very much out there. Studies in the US and in Europe have found almost across the board between pythons around a 20% prevalence. So it's out there. And then really it's about managing the circumstances that pathogen can actually cause disease, because it would, and I'm going to, not anywhere near as versed on crypto, but I would say, at least as it pertains to serpentivirus, it would actually be a pretty very small minority of positive snakes that actually go on to develop full-flung, life-threatening type illness.
With crypto, it's the same thing. The number of adult snakes that have it is astronomical. It's something has to happen to that animal to lower its homeostatic state, to make it basically sick. And then that's when it expresses disease. But that animal could have had crypto for years. And every time it defecates, it's shedding spores. So, these pathogens that kind of have this boogeyman effect with people, they're far more prevalent than people realize.
Mm-hmm.
Yeah, that's the deal. So you have to almost kind of look at this from a biological, philosophical point of view of like, well, how many asymptomatic carriers do I want in my collection? Do we just torch them all? Do we not torch them all? If it's not expressing disease, it is disease, but is it actually, harming my collection? Yeah, these are the kind of questions that I think people don't really think about and should, because it's just naive to assume that if you have 100 ball pythons, that you don't have some virus lurking in there.
Sure.
Now, I will, and so I'm going to walk a pretty fine line here and say that, like, what I'll talk about next is just kind of my own personal thoughts, having worked with snakes for the past over a decade, and then kind of matching that with what I've seen in a lab. And I can narrow down a set of conditions that seem to commonly occur when we see outbreaks on like the collection scale. You're kind of talking about, it's a big group of viruses. They have multiple different potential life strategies of how they want to replicate. What I have seen is that a lot of times when you get this big collection killing virus, it happens when you try and merge adult animals into an existing adult animal colony. And it seems like there's kind of asymptomatic virus probably native to the colony and potentially a new virus that you're introducing. And in both of those colonies independently, those pathogens didn't do anything. But, you know, maybe the cross reactivity between the actual individual snakes is not very high, where, you know, one strain in one colony does nothing, but then you immediately drop it into another colony, and that's where you can kind of have problems occur on a wider scale. And then I would say the other thing that seems to be commonly seen in the recipe that ends up in a bad virus would be like if you have a chronically sick snake. You know, if you have a snake that is just sitting there with an RI and it's not really getting better and it's not really getting worse, at this point in my head, that is a virus mutation factory. And I am very heavy-handed in terms of removing those animals from a colony at this point. So I will say that you know, herpetoculture is still here. These pathogens probably themselves aren't anything that new, almost certainly the case. You know, now we just have tools in our kit to be able to test for it and manage it. So it's really kind of the extension of a existing story and not, you know, the world is ending. You know, if you go through pretty basic biosecurity of being cautious about bringing stuff into a colony, of managing stuff in a colony when you do see problems, especially if you're buying neonate animals, that's a pretty safe route to go. You know, there's things you can do that really hedge your bets into not ending up with problems, you know, before you even do a single test.
Sure. Just going back to the first part of what you were saying. So you mentioned you're having a group of adult animals that has one strain, and you have a group of animals that are also adults that have a different strain, and they're both okay on their own. And then you put them all together in the same room. Well, strain B therefore affects A snakes, and then A strain affects B snakes, which causes them all to crash.
That would be my, you know,
just so that I'm laymanizing, you know what I mean?
Yeah, that would be my hypothesis on a good way to end up in a dangerous
that makes
spot.
sense.
Yeah, we kind of touched on this, you know, like a year or so ago when I was on with Dr. Porcher.
Mm-hmm.
You know, like Steve said, there is so much diversity within these viruses, and with that diversity, you know, part of that is very likely, you know, genus specificity or species specificity. And so you will have some of these strains that fundamentally are benign, but they're benign in the species they have, you know, essentially co-evolved with. Or they're benign for some other species because the jump is just too far. You know, there are colubrid serpentaviruses.
Right.
They don't seem to do anything to the colubrids, and they also don't seem to cross into that, you know, that boid barrier either, because the difference is just too far.
I would definitely even go. So this kind of reminds me of some of the cell culture stuff that I do, which we try and isolate these viruses so we can actually work with live virus in the lab to really experiment with it. But some of these viruses are super, super selective on what they'll grow on. And it's really interesting. You know, I had a Burmese python virus, and I literally couldn't grow that virus on Burmese python cells. Yet I have this ball python virus, and it's a serial killer and will totally wipe out any cell line I put it on. So, you know, it's a big umbrella that we're talking about here. And part of what's significant is the individual virus that's at play. Part of what's significant is, you know, the actual colony, how it's managed itself, and the individual animals and their resilience within that.
Yeah, I remember there was some time back before any of this was relative, there was an issue with a lot of the big box pet stores when maybe 10, 15 years ago, when, you know, ball pythons and red-tail boas had really boomed in terms of people that aren't herpers, you know? And I remember there was a thing with, I don't know if it was Petco or PetSmart or Pet Supermarket, and they basically said, well, the husbandry is the same for a ball python as it is for red-tail boas, so just keep them in the same enclosure, you know? And both snakes die because they're drinking from the same water bowl and they're swapping spit. And who knows what the real reason was for that, but all the other snakes in there were fine. And if you had a ball python in the same rack as the ball python that died with the boa, the ball python was fine, and vice versa. So it makes sense.
Yeah, and some of those, that could have been some IBD stuff. I remember when IBD was, what was probably about 20 or so years ago. We basically saw the same thing with IBD that we're seeing right now with Nido. Everybody was panicking, everybody was freaking out because
and everyone had boas.
Right. Everyone had boas, and oh my God, all these boas, they're stargazing. There's all these problems. And boa keepers started picking up ball pythons to use basically as canaries in the coal mine because IBD is generally super lethal to pythons. So boa guys would expose these ball pythons to their collection, and if the ball python died, then they'd know that they had IBD lurking in their collection. Now, as time has gone on and we have found there are some pythons that are actually kind of resistant to IBD itself, and it's not always just an absolute killer. There are some pythons that they reach that same static state with IBD, and they can persist and carry on with it. And again, that could be some fundamental change in the virus that they were specifically exposed to, or it could just be a survival of the fittest thing where you found a python that just wasn't susceptible.
And maybe that's where a lot of the confusion lies, at least for myself. And I know I've raised this question to P and Cody, and in pertaining to Nido specifically, is okay. I've got a positive animal. It's completely asymptomatic. There's no issues with it whatsoever. I've had it for years. I haven't had any problems. But now all of a sudden I need to either euthanize it or completely separate it for the rest of its life. Same with crypto. If it's as prevalent, obviously people aren't losing their collections every day. People aren't having mass die-offs or anything like that. And so it's kind of one of those things where people are like, yeah, it exists, but nothing's happening. So what? And I don't. The question is, I guess, is okay, it's there. So now what?
Well, and I think what we will probably end up seeing is sort of a balancing effect. Like, I think we've seen it with crypto in the colubrid world. They've sort of just embraced the idea that, crypto is there. And most people understand that crypto is there. That's accurate, right, Zach?
Yeah.
Like most colubrid keepers know that. And they try to treat it, and at least most of the ones that are going to be honest about it will try to treat it and regulate it and keep it under control in their collections. And if they have an eruption, they're not, sending out all of these sick animals.
Right.
But you're also not hearing about these massive eruptions because the colubrid world knows that they happen. But the python world is only still like coming to grips in the early end of, oh my God, somebody just lost their entire collection to Nido. And, this happens, but this happens probably for a bit of a reason, like Steve said, somebody merged two collections, or, something has, somebody went away for a month and they're, the guy who was taking care of their collection wasn't taking care of them to quite the same level. So when they came back, there had been enough of a change that the animals slipped just a little bit in their health, and it allowed the virus to amp up enough through the collection that it just, boom, and it melted the collection down. And then you hear about it and everybody in the python world freaks out. And, this scare factor thing, yeah, it's scary, but it's not always like this. It's not always the end of the world. And, Justin, you said it great, is I have seen so many sad cases of people saying, I tested my animal and it came back with Nido, so I had to put my whole collection down because I didn't want to test all of them, but I have to assume they all have it. And it's like, No,
throwing the baby out with the bathwater.
Yeah, it is. And it wasn't
Yeah.
just, they tested a new animal that they just got in, and the animal was outwardly healthy in all appearances, Now, one, I have to say, why wasn't that animal quarantined that you suddenly feel you have to destroy your whole collection? But two, if the animal is outwardly healthy appearing, and now you know that it's tested positive, if you are maintaining good biosecurity practices, if you're not throwing all of your pythons in the same tub as you switch their media out and their substrate out, if you're not just taking the rat that you slammed in that sick one's face and it doesn't eat it, and then so you go slam it in another one's face, if you're not doing those things to spread the disease, then your other animals are probably safe. And so, don't throw the baby out with the bathwater.
Right. And
if you want to test and you find your animals are positive, that then becomes a matter of, I need to monitor these animals. I need to keep a closer eye on them. I need to maybe up my husbandry care on them. But if they're not sick and dying, why are you freaking out and panicking? The same way, if 50% of colubrids have got crypto and they're not all dying, why aren't we all panicking and killing all of our colubrids off?
Right.
Go ahead,
real quick, because I don't want to lose on this particular thing. So I'm a good example of this. So with my non-venomous stuff, I do quarantine. With my venomous stuff, I'm physically not allowed to have a quarantine per Fish and Wildlife because I'm only inspected and certified and verified by Fish and Wildlife for one room for the venomous. So that means that if I bring in new venomous, it has to basically just go on a different wall. It's better than nothing, but I don't have the physical ability to do that. And that's something that I acknowledge, and that's something that I understand. And if I bring home a baby rattlesnake and it kills off the whole room, that's something I have to deal with, because unfortunately we have laws, we have rules and regulations, and even though sometimes they don't always make the most sense, they are there for a specific reason. That being said, though, because I know this is a possible factor, I do the reverse, where if I am going to get rid of or sell or gift or trade one of those venomous from that venomous room, it goes into reverse quarantine, where now it's isolated to make sure that before it leaves, then it goes through the proper rigor so that I don't infect someone else's thing. God forbid I have something I don't know about. So I think it also comes down to, if you're going to quarantine, great. If you're not going to quarantine, great, but acknowledge what you're doing and be honest with yourself. You know what I mean?
Yep.
What were you saying, Dr. Loughman?
With the crypto, so as we all know, colubrids have a bazillion different communities. There's the hognose community, the corn community, the milk community, the lamp community, garter community, like just tons.
Soma.
There you go. And I do think there's still an element of panic in the colubrid community. The community that's had to deal with this the most is the hognose snake community. And what's really weird about hogs and crypto is that there's Cryptosporidium serpentis, and hognose snakes can West Plains hognose, Western hognose, whatever you want to call them. They can get serpentis, but the one that seems to really do a number on them is Verinae, which is the lizard based Cryptosporidium. And what's been rather interesting to me is that if you produce snakes and then sell a snake and then somebody tests it and it comes back as crypto, you're going to get a scarlet letter. And that's understandable to a point, especially if you know that you have it and you're hiding it. But the assumption always is that the person knows that they had it. They're just hiding it. And I think that that's part of this. If we're going to deal with disease, you kind of have to, everybody needs to put their big boy and big girl pants on and understand that people can make a mistake, and it doesn't mean that they're the worst herpetoculturist known to mankind. I know a lot of people will actually, if you tell the breeder they have crypto, there's mass panic, and then they'll move forward. The thing that
pathogens do not care if you think you're doing everything right.
They don't. They do not care. But the thing that's horrible about Cryptosporidium, and I'm actually curious to hear if this is the case with serpentivirus, is the vector and the pathogen load for a snake to become infected is ridiculously small. So with Cryptosporidium, as few as 5 spores ingested through like drinking, and now the animal has it. And given the crazy life cycle that Cryptosporidium has, they do a lot of asexual reproduction. So basically the snake ingests a single spore, and then that single spore divides and is now 2 spores, 4 spores, 8 spores, 16 spores, blah, blah, blah. And so that's why it doesn't really require that much for the snake to become completely infected. But one of the main vectors is you can like try to be as sterile as humanly possible. But if you have flies, forward flies, every time a snake goes to the bathroom, a forward fly goes to the poo and gets those spores on it. They are a vector of this disease. So like in that situation, you can try to have a crypto-free collection, but you have a forward fly, you're doomed. So what we did at the university, what some people have done, is they basically will test their snakes, get crypto positive snakes, and then have we call it a crypto ward. And so basically there's an adage when it comes to diseases where like space equals safety. So and doors are a biggie. So you basically go up 1 or 2 floors and then put as many doors between as humanly possible. And that's a way to kind of have your crypto snakes and your non-crypto snakes. But I also understand there's probably people listening to this who are like, why the hell would I keep a crypto snake if you're telling me 5 spores and a fly and the whole collection is going to get it? But the thing is, if you test that, if one of your animals tests positive, it's likely you've got more asymptomatic snakes. So you know this really involves some soul searching and some understanding of basic disease biology to kind of get past the whole, just because your snake tests positive for one of these viruses or Cryptosporidium or another pathogen, it doesn't mean you need to burn the house down or nuke the collection.
It is definitely nowhere near as simple as animal plus pathogen equals bad time.
No. Great example. If you have the flies and you get rid of all your snakes and you're going to start over again, those flies are going to basically perpetuate the disease. And so you have to also get rid of the flies. And the flies can live in your drain, like in the elbow of your drain. You can have a forward fly colony surviving there. Nobody thinks I have to clean out all my elbows and all my drains in order to get rid of this disease. And then you bring the animals back and then boom, you've got crypto again. So like it's a learning to live with it and deal with it is much better than trying to obliterate it. It's a much more realistic approach.
Well, I mean, the whole point of oh, go ahead, Steve.
In terms of making that same sort of parallel for nidoviruses or serpentiviruses, we really don't know what the viral load is to infect a different snake, and there's so much variety that it's likely to be different for each one of these things. But it definitely seems to be more likely to spread in directly adjacent animal enclosures. So it seems like for at least some of these, there needs to be a pretty good amount of contact between animals to really pass along that pathogen in a lot of circumstances, but nowhere close to every circumstance. So it's hard. I don't know that I have a fully fleshed out end game of what I think a entirely responsible herpetocultural community would look like dealing with these pathogens. It's still, I would say, a very much unwritten book, and because there's just so much variation that I really think you just do the best you can with the knowledge you can, and hopefully that'll inject some forgiveness in the hobby to really kind of understand that pathogens don't automatically equal bad seller. Again, you can do everything right and still end up in a spot where you never thought you would. So
Yeah, I mean, Erica Paris is a really good example of that. Like, she cut off her collection. She tested stuff, did her best to make sure that she had nothing coming or going. Still had Nido. Like, Nido still popped up even after she had stopped bringing in, I think it was chondros. She did everything possible. Like, she really went above and beyond to make sure that that wasn't a problem, and then still got a positive animal out of it.
Yeah. So I have 2 buildings that I work out of here, and I kind of within those buildings have 2 different strategies for how I'm dealing with nidovirus or serpentivirus specifically. So the one building has a lot of blood pythons in it, and I call that my virus neutral colony, where every single animal that's in there has tested negative 3 times, and yet every single year I'll get one animal that'll get stressed enough during the breeding season to pop with a respiratory infection and then test positive. So that's been a closed colony for almost the entirety of those animals' lives. Like, the sequence of that matches the blood python virus that I knew was present when I had seen disease in the past. And so, here's an example of a colony where there's virus in there somewhere and I can't find it, because if they're happy animals, they're not going to test positive. If they're not stressed, they're not shedding enough virus to test positive. So, I don't really know what the best way to navigate that type, that kind of colony is. But, we don't see vertical transmission. So if I'm really paranoid about it, the easiest thing I can do is just hatch my babies off in a different building or keep them in a different room, which is what I did with my second building. And so this second building, I really wanted to take as biosecurity as kind of far I could take it in a hobbyist type scenario. So it has, it also, because I'm in Florida, I needed the double entry anyway. So I turned that into a airlock with foot bath and foot wash and, sanitizing protocol with LED indicators to determine when you can, you've waited enough time for the contact time to come in for the sanitizers. It's just so ridiculously over the top in terms of precautions I put in place, and every animal that is in there is stuff I've tested 3 times as neonates. So, should theoretically be the safest possible configuration for a colony. And I will be surprised if I can truly end up in a point where I can say even that biosecure building has no serpentivirus in it. Like, it just, it's there. But even with the best you can, I don't really expect perfection.
Yeah, and I mean, that kind of goes back to the question I asked earlier, is like, if that's the case and you now have positive animals but they're completely fine, like, what do you do? It's like, that's my biggest issue with the whole thing, is like, I have a ton of corn snakes. I could have crypto. I haven't tested, and I'll go ahead and throw out sort of where I stand as far as that goes. Myself and Billy Hunt, I think Phil is kind of in that boat too. Like, unless the snake is outwardly sick and there's an issue that is not in the norm, like there's something that is clearly not, that is out of what that baseline for that animal.
Yep.
I'm not going to test unless I have unless I feel like I have a real reason to. Like, if something comes up, chondro stops eating when it's been eating solid for years, all of a sudden stops, it's not in a shed cycle, there's no other issues, then I'd be concerned. Then it's like, okay, something's up. But then I'm going to go through other potential areas where that would
troubleshooting
cause of, yeah, exactly.
Right.
But I will test. Like, I have no issues with testing, but it's not like I'm not going to go test every single animal in my collection because I'm scared that crypto might be there.
Yeah. Yeah, and let's also say that just because a snake develops some kind of RI doesn't mean that it's serpento, and doesn't mean that it's going to kill everything off.
Yeah, so if we look at statistics, there was a paper out in 2019 that, basically directly taken from the paper, despite evidence that serpentiviruses are common and are potentially significant pathogens, the extent of susceptibility to infection and disease remains poorly understood.
Is that the one that's longitudinal cross-sectioning?
Senglin. I think it could be the longitudinal cross-sectioning.
Yes. Mark D. Senglin.
Yeah.
Okay, so I have a few of those graphs that I have on this on the screen in case you guys wanted to use them.
But there was another one from last year, Schlatu. They found that of almost 400 positive animals, 400 Nido positive animals, only 75 of them actually showed symptoms. So that's like a 20% rate.
Mm-hmm.
Yeah, I can.
But the flip side is also that out of 900 animals that tested negative, 130 showed symptoms. So that's like a 15% rate. And from that paper, they specifically say there is no correlation between symptoms and finding serpentavirus positive, establishment. So that goes to Justin's point or Phil's point of just because your animal is sick doesn't mean it has Nido. And you'll see that a lot too. Like, people are like, well, my animal died and it was wheezing. It was Nido. It's like, well, did you send that animal in for pathology? Or did you just assume that it was Nido because Nido is the hot button thing right now?
Mm-hmm.
I mean, it could have been paramyxovirus. Nobody thinks about paramyxovirus, but that's a huge killer.
Yeah.
And it causes basically the same symptomology that we see in Nido. It causes respiratory distress. It causes mucus discharge. It can cause neurological symptoms. So you could be having a paramyxovirus outbreak in your colony, and you just blame it on Nido because that's the hot button ticket right now.
Right.
But I mean, in y'all's opinion, is sort of where I sit with that. Is that an outlandish place to be in terms of the whole thing? Like, honestly,
I mean, I think it's basically
don't test unless it's sick, right?
Yeah,
right.
I think that's basic. I mean, I look at my reptiles the way I would look at basically any pet, with some slight caveats.
Right.
I take, when you've got a dog, you pretty much the same way you do with yourself, you take your dog into the vet once a year for a wellness visit. Now, part of that's also because you've got those regulations that say, you have to have your dog get his rabies shot every year, but aside from those annual tests that you do just for wellness, when do you take your dog or your cat or your horse to the vet? You take it when it's sick. Okay,
when it's acting weird.
Right. So that's the same thing. I'll take my snake and test it when it's acting weird, when it's acting sick. Now, there's one other time when we very frequently take our cats, our dogs, our horses, our rabbits, our whatever in. That's when we first buy them. You go to a breeder, you pick out the dog that you want from their litter, you get that puppy, and what's the first thing you do? You take it to the vet. And it's a wellness check, but it's a start off wellness check.
Right.
And that's how I see a lot of, but I can see that as a direction of, these, I just bought a snake from breeder X and I tested it for Nido, or I tested it for IBD, or I tested it for crypto. That's that first wellness test. Now, if I bought that dog and I took it into the vet and the vet says, your dog has parvovirus, then I would go back to the dog breeder and say, I got parvo. Your dog has the dog that I just bought from you has parvo. And a reliable dog breeder will say, okay, we need to make this right. And to me, that's, that was the legitimization that I saw John going for with this morph market policy was, let's bring the reptile hobby into the same legitimate sphere that all these other pet industries have.
Mm-hmm.
Like it should be.
Right. And, so it was very discouraging for me to see big names and well-known people thinking that this was a terrible, horrible idea, because it's like, now wait, you're supposed to be one of these, these preeminent breeders. If somebody came to you and said, I bought an animal from you and it tested positive because I did a wellness visit, why wouldn't you act like, do
something about it,
right? A dog breeder or a snake breeder. Now, there are some breeders out there that will. I didn't see anything. I didn't see and I'm just grabbing names. I'm not throwing people under buses or anything. I saw a lot of chatter. I didn't see all the chatter. Obviously, I didn't see Kobilka say anything, But I know from a personal friend, he bought a snake from Kabilka. He tested. It came back positive. He called Justin and said it was positive. Justin said, I understand. Send it back. I'll refund you. So there's that legitimacy. And I'm glad that Justin was willing to do that. And I found out later that Justin tested that animal when he got it back and it came back negative. And then this comes down to that everybody's like, well, all these tests are unreliable. All these breeders are saying all these tests are unreliable because people get Nido positive tests and they're not really positive. I would say people don't understand the difference. Well, not necessarily difference. I don't think people understand the positive, the negative, the frequency of false positives, which is, while it's not an impossible thing, those are kind of low in well-designed tests. But they're also dependent on the nature of the test. And if, the buyer sends it to one lab, but the breeder ends up sending it to another lab, that could account for why you get two different test results.
Right.
Yeah, absolutely. It could. Yep.
Let me ask this too, since we're on this real quick. Is there, because I'll be honest, I've never tested anything for Nido. On these Nido tests, when you get your results, is it cut and dry like a COVID test where it says positive or negative?
Yes.
Or is it a percentage, much like some of the other like human illnesses where it says, okay, between 1 and 4 is you're sick, but the test goes to 10. So if you're a number 10, you're fine, but if you're number 4, oh, you're sick. Like, is there a scale to this, or is it just yes or no?
Steve might be able to better talk on this, but from the type of testing that I will do, I work a lot with quantitative PCR tests. Those, they're not necessarily binary, but they're often used as a binary. Usually a qPCR test gives a score back that's kind of high or low. There's a threshold level that we mark as this is positive. If it's anything below that threshold, we call it negative. In reality, it may not be truly negative. It could just be that whatever is there it's such a low volume that doesn't hit that magic threshold that we put down.
It's so low it doesn't really make a mark.
Right. There are other tests, more basic, just PCR tests, where they're binary in as much as you're not looking at a kinetic time frame of how it happens. You just look at the end point, and there's either a signal that says there is a positive signal here, or there is not a positive signal here. And again, if there's not enough there to get that reaction high enough, it'll say negative, even though it's not truly negative.
So the reason I ask is because I could see several people in our community who are not necessarily, I don't want to say they're uneducated, but who are not as savvy to it, much like myself. Like I said, I've never used one of those tests. I have no idea. And I read the manuals. I'm not one of those guys that doesn't, but I could see where someone sees that percent scale and maybe reads it incorrectly and overreacts, or reads it correctly and underreacts. So that was the reason for my question.
Well, is it the same with crypto too? I mean, when you get a crypto test back, what are the results usually? Is it just a positive or negative?
Normally positive, negative.
Does it denote the
and then it denotes
snake versus lizard?
Yes, and you can with the most frequently used lab, it will tell you the species. So, and that's through basically phylogenetics. They're putting it into clades and all that kind of good stuff.
Yeah.
The problem with crypto is that the damn spores have this, for lack of a better word, spore coat around them that is extremely difficult to get to open up to get the DNA out to actually get the positive. And so that's why I've talked to a lot of people about crypto. And I'll say you got to do it in triplicate. Just because you get a negative doesn't mean you're in the clear. Because what could happen is with a crypto sample that you're sending off to the lab, most of the time what people are doing is they wait for their snake or lizard or whatever to go to the bathroom, and then you take the swab and then you swab it down in the feces. If you swab the urates, there's going to be spores there, but unless it's like complete liquid crap, there's probably not going to be that many spores there. And so then you've swabbed the wrong part, or you swab the feces itself, and as that bolus of poos leaving the animal, certain parts of the gut are going to have more spores than others. And if you swab the wrong part, you may actually somehow not get that many spores. In an asymptomatic snake, that's the deal. If it's a symptomatic snake and you have the pure diarrhea, it doesn't matter where you put that swab. You're probably going to get enough because these things are shedding millions of spores. I mean, the number of spores that are leaving is crazy. In a asymptomatic snake, it might be shedding hundreds of spores. It might only be like 150, 200 spores. So if you don't get the spore on the swab, you're not going to get the positive test. And then the lab has the hard part of actually getting that spore to crack open, which
and for
is
an analogy
very hard
there,
to do.
Yeah, for an analogy there, like if you think of the living modal active crypto organism, it's basically like a tomato. You could squish it really easily with your lab techniques, but the spore is like a coconut. And when I say like a coconut, I don't mean like the little brown one you get at the store. I mean like the one that's got a giant green husk around it. If you want to get into the center of that, the force that you use to squish a tomato is not going to do a damn thing. You need a jackhammer to get in there.
Right. So I think we could probably use this as an opportunity to kind of contrast between the two. I know, crypto is incredibly resilient in the environment, whereas these RNA viruses, in contrast, are very much not. So I can kind of go into my research, but I guess before, if you want to like set the baseline of what it takes to truly decontaminate a room or equipment or a colony that has crypto, these spores are incredibly resilient.
Yeah. There's a lot that circulates as to what people think will kill crypto. And I've said this on the THP with you guys. I said it on From the Ground Up back in the day. I think I mentioned it a couple times on my podcast.
Right.
With Cryptosporidium, there's basically 3 things that will kill. Well, we don't even use the word kill it technically. The terminology is deactivated because it's an apicomplexid protozoan, which if you really want to learn about, if you want to learn about an organism that's as close to an alien as you can get, just do a deep dive on apicomplexa. I mean, they are nuts. But the thing that gets through that coat is 12% or higher hydrogen peroxide. That will kill it. That is not the hydrogen peroxide you buy at the at the supermarket or the drugstore. That's 6%.
Yeah.
And that tickles it. Like, it's just kind of like, ooh. And that's the extent of it. I mean, you're not hurting it, killing it, anything. You might kill off a couple weak individuals, but you need the food grade is what it's oftentimes called. And you can get that on Amazon.
And that's not that harmful to us. Like, it's not going to cause like
no, it's harmful.
It's harmful.
Okay.
That's use gloves. That's be in a very well ventilated area.
Yeah.
Okay.
Like if you get that on your hands, like
you turn
if you've
white.
ever used just the home stuff that you buy at Walmart,
right,
and you use it for so long that the finger ridges get white. Yeah, the 12% stuff, you spill it on your hands and you get that white burn like that.
And if it goes up into you breathe it in, your mucus glands start to dry out almost immediately, and it stings and burns. It is not pleasant stuff.
Yeah.
The other thing that will kill the spores, which is really interesting to me, is sunlight. So, but we're talking like if you have a high UV index day, one of the best things you can do to get rid of the stuff is to hit it with the 12%. Hydrogen peroxide, and we'll talk about what that means here in a second, and then immediately take the enclosures outside and put them into the sun, and then really cold temperatures like minus 10, 20, 30 below zero will also deactivate the animal. But like all the bleach does nothing to crypto.
That's wild.
Ammonia will kill it, but the spore has to sit in high concentrated ammonia for 12 hours. And who the hell wants to do that?
Oof.
The kill time, by the way, for the peroxide is about 20 minutes. So that means that it's not just you put it on and you're dead. And I'm sure Steve's going to tell you, like, that's kind of how the viruses will work. But with the crypto, it's you're hammering it, and the stupid spores have to sit in the liquid for up to 20 minutes to then deactivate. So that's why, like, just sunlight works. But the problem with cryptosporidium, and this is the biggie, is that, remember, we only require like 2 to 10 spores for the snakes to become infected. And if you think, I got PVC enclosures all around me right now, and when you screw those things together, you've got that little gap contact point where they come together. And we've all done the thing where you wash your enclosure and then the urate, poo, whatever kind of goes to the edge and then goes down into that crack. You can have the spores just chilling down there and think, I cleaned out everything. So to really, I mean, if you're super paranoid and you absolutely want to make sure that you've wiped it out, and you still can't really even get to that point, but you can get as close as possible, you may want to actually contemplate just disassembling the PVC enclosure and just laying it out in the sunlight. Contact time for UV, I think, is like 10 to 15 minutes. It's not that long. But you know, that's it. I know that killing off and wiping out crypto through sterilization it's a nightmare scenario. I literally, when we had our big outbreak in, I think it was 2018, that's what I did my entire spring break. We disassembled. I didn't go to like on a field trip or anything like that. I got a Tyvek suit, prepared me for COVID very nicely. Bought some steam cleaners, a whole bunch of hydrogen peroxide, and then basically took apart 20 PVC enclosures. Conveniently, we had a polar vortex happening at the time. So we steamed them. We hit them with hydrogen peroxide. We then put them out in the bright sunshine, and it was -20 outside. And I do think that we actually that's as close to a complete kill as I'm ever going to get. But I'm not going to say it was a complete kill.
Yeah.
So.
Yeah. Now, let me ask real quick, since we're on the topic, if let's say I get crypto and my entire collection dies and I have no animals left, but I clean out all the cages with 12% and I don't do the sunlight thing because I don't have any animals. They're dead. What do I care? And 6 months later, I say, you know what? It's time to buy another snake. How long are those spores going to live in that cage, or even the protozoans themselves, before they're dead and gone?
How long can they lurk?
Right.
They
exactly.
can lurk better than the best lurker.
Okay.
We're talking. Nobody really knows the answer to that question because no one's done a study long enough to figure it out.
Wow.
But they can live years. I mean, that's the whole point of an endospore. These animals, if you think about a parasite, because this is a little bit different than a virus. When it comes to a eukaryotic parasite, their main goal is to get out in the environment, and then they have to persist in that environment long enough for their host to come by, serendipitously, by the way, because no colubrid wakes up in the morning and is like, you know what? Crypto today. That's what I'm going to get. They just don't do that. So that's why the organism basically manifests itself and does the asexual reproduction and all that craziness. And then when you have the active pathology kicking in with crypto, that's when the snakes are shedding millions of spores. And then what's even cooler, and if you're a good biologist, you look at these things, and even though I hate crypto, I hate what it did to my collection, but I really respect the bastard. I mean, it's a pretty cool critter.
Yeah.
The part of the life cycle corresponds with times of physiological stress. And if you think about like temperate snakes, one of the major stressors is coming out of hibernation or brumation, whatever you want to call it, and breeding. And that is the best time, if you're a crypto organism, for the snakes to be shedding you, because this is when all the snakes are together.
Yeah.
And so they don't want to do that just necessarily like in the middle of brumation. They want to hit them when they're stressed, and sexual reproduction causes stress, physiological stress. So that's probably not an accident that's part of the organism's biology.
Right.
And so we see crypto manifest itself in people's collections with colubrids 2 times a year is when I start getting all kinds of messages. When you're putting things into brumation and you're cooling them down, that's a stress-inducing activity. And then when you're pulling them out of brumation and then you're putting your pairs together, that's stressful. And so then you see disease take off.
I would say that general rule also applies to the serpentoviruses or nidoviruses, which you can kind of bake into your testing strategy, because if you have those low-level positives and you have a time of year where you know your colony is stressed, that's a good time to maybe pick up one of those low-level positives. But yeah, I'm glad I had.
Yeah, that's how I would say that's how pretty much any pathogen is going to work in some respect, is when there is susceptibility, that's when they are going to be at their strongest, because that's when they can replicate the most, because the animal's body is not going to respond and keep them in check as well.
And that's probably why in November you keep hearing, carpet guys, man, that one female's blowing bubbles again.
Seasonal
But again, that may not be just Serpento. That could be Paramyxo. That could be Rio. That could be less likely to be Adno. But I mean, it could be something else.
You could also.
I just think that everybody freaks out over Nido because it's the hot button right now.
Right.
I mean, you also have opportunity. It's not even. It can get more complicated than that, because you can have a stressed out snake that has a low level infection that's upticked a little bit higher, and because that immune system is dealing with that, then you have an opportunistic bacteria that jumps on the train there and really takes advantage of. None of this is easy.
Well, yeah, I mean, that goes back to what you said at the beginning, where when you're trying to make cell cultures, you pick up all kinds of just random crap. It may be Nido, but it may not be that the Nido is causing the disease. It's that the Nido ticked up enough that a normally commensal bacteria, you've got a pseudomonid or something that's causing an active lung infection.
Yeah. It could be anything. It could be a gum infection because it bit the prey item wrong and got a tooth stuck in its own face, and honestly that spread and went bronchial, or whatever.
So
Conjurers are dumb. I've seen them do stupid things.
That's it.
I'm glad I had Zac go first, because it's way easier to deal with these viruses in terms of like the environmental context. In terms of contrasting what a nightmare it could be if it was something more along the lines of crypto. These being enveloped RNA viruses, they're pretty easy to break up, and once they're broken up, it's pretty easy to break up that RNA. Like, they're really not designed to be tough things long term. So a lot of my research is taking this cultured live virus and then manipulating it through temperature or any number of various sanitizers, and then kind of seeing how much live virus is left in that sample. And so from preliminary data on that and based off of related viruses, it looks like outside of 10 days, there's pretty much no active virus left outside of the host. And even within that, that virus starts to degrade pretty quickly, where even after a couple of days, you're seeing massive drops in the number of live virus actually present in that sample. And likewise, they're pretty easy to kill off with sanitizers. So I've done preliminary research on bleach and ethanol, and both of those knocked these viruses back, unsurprisingly, no problem. So you can kind of hypothesize that UV is going to be pretty equally potent, and that there should be a whole suite of sanitizers that will probably help quite a bit, at least certainly in comparison to if you're dealing with trying to sanitize equipment after crypto. So
only one I would hesitate on, and this is purely from extrapolation, just from other somewhat related viruses, would be chlorhexidine, because I know that some of the other viruses in the Nido family, some of the coronaviruses, some of the other toroviruses, are a bit more resilient to things like chlorhexidine. But again, that is pure extrapolation.
Sure.
But I know a lot of people use chlorhexidine in their collections.
I've been using Zep.
Yeah, that you literally took the words out of my mouth. I was going to ask you, gentlemen, is I use the blue 2% for my entire life, and everyone told me, oh, well, red 5% is way too abrasive. It's way too harmful. And now I'm kind of like, well, blue doesn't do anything, I might as well go to the red. So
I switched to Zep. That's what I've been using. I don't know if y'all have done anything with that. I get it at Tractor Supply. It claims to kill viruses and kinds of stuff.
I have no idea what the active ingredient is.
Yeah, I'm not sure either.
If you're not trying to get through a spore, bleach is really just not good for living things.
Yeah.
I have a PhD, by the way.
Yes.
Like, I mean, that just kills stuff. Like, I mean, I remember being in a microbiology class and they explained how all this stuff worked, and it was very, very made an impression on me that you can. I'm not saying don't use the fancy sanitizers, but if you're like, if you're on a budget, bleach works. I mean, even Dawn dish detergent is really good at taking those envelopes and just basically ripping things open and exposing the nucleic acids and killing stuff. So
very interesting.
The active ingredient in Zep is a quaternary amino compound. So it's a cationic surfactant. So it should break up a lipid envelope. Yeah, I know. It's all chemistry. That should disrupt the viral particle envelope on these envelope viruses.
Yeah. I mean, I guess contact time is important to mention that, it's not like dab it and it's good, but within reasonable use, these are not tough things.
And I working in a lab where we deal with a wide variety of molecular organisms or microbial organisms. When we're cleaning our hoods out, our contact time for bleach is 10 minutes. And that's 10 minutes wet. So like if you just take
undiluted, right?
It's 10% bleach.
Okay.
So if you take 10% bleach on a paper towel and wipe it down and it dries in 60 seconds, that's not going to be enough contact time. What you should do is make up like a 10% spray solution and spray whatever you've got down and leave it damp for 10 minutes and then wipe it off.
Okay.
Okay, so I swear to God, I'm not wearing my tinfoil hat, but ivermectin for the virus in animals.
No, that's an anti-helminth.
I mean, I've used this stuff before. I know it's like for mites as a solution. Okay,
right.
Preface: I'm not injecting animals with straight ivermectin because that stuff is really gnarly.
Ivermectin is very gnarly. It's an anti-helmintic. It does work against mites and things at certain doses, but again, really gnarly. Good way to potentially poison the shit out of your animals. So in very, very broadly done, not very well repeated studies with some viruses, ivermectin was shown to have an almost insignificant but slightly positive contribution. Now, it got a whole lot of, and I hate the fact that we're bringing COVID up because it gets so political and tying politics into snakes is just
brought
stupid.
Up in the chat. It was a question.
Yeah, we're going to keep it straight. Don't worry.
Yeah, but the studies that linked ivermectin as helping COVID have all been proven to be horribly, horribly flawed. And further studies have showed that it is not helpful and is in fact potentially detrimental, mostly because people are then just mega dosing on the stuff and poisoning themselves. And so don't go giving ivermectin to your snakes thinking I'm going to cure the nido, because what you're going to do is kill your snake from the neurotoxicity of the ivermectin.
And it ain't a quick death either.
No, it's gnarly. It's
that
miserable.
Works great, but oh my God, can you overdo it so easily?
It gives them neurologic problems. It gives them hepatic problems. It destroys their liver. It's horrible. If you have ever seen a snake get overdosed on nido or toxified, or not nido, by ivermectin or toxified by ivermectin, it is a very unpleasant experience to see.
Yeah, I don't even use it on mites anymore now. I switched to frontline spray, and that works light years better. And I don't have to worry about killing things.
Yeah.
Outside of the mites.
So just trying to get us back onto the train track, so to speak. So we covered basically concepts of when one might want to test, when one should or possibly doesn't necessarily need to test, and what goes involved in testing and keeping your eyes open to what your critter's doing, right?
Yep.
Um,
I think one thing to add to that list is
Sure.
oftentimes when people see the manifestation of perceived disease, so you have, I can speak for colubrids, so you fed your Florida King a pinky, and then you come in the next day and it's regurgitated the pinky. A lot of times people go straight to crypto, and like, could just be husbandry. It could be cold. Like, I think that it's important for people when they see the disease, what they perceive to be disease, to kind of take a step back and almost start running through what we call null hypotheses, which is basically, all right, I think that it's crypto, but what are the other things it could be that led to this conclusion?
Right.
And
Don't
I know,
let your hypochondria take over.
Yeah. And I can use myself as an example. So I picked up a pair of blonde subox in July, and they just got through their 60-day quarantine, and I brought them into the main collection and I started feeding them, and love my subox. Both of them regurgitated. I was like, son of a bitch. And I broke my own damn rule and was like, oh God, we've got crypto. And the panic ensued for about an hour. And then I calmed down and started looking like what could have made them regurgitate beyond that, because they didn't have any other symptoms. They didn't regurgitate during quarantine. They didn't ever have liquefied stools. They didn't have the swelling in the gut. Like, there was nothing other than the fact that they had vomited up their mice. And then I looked at the rack that I moved them into, and it's cold here, and I think that my thermostat probe crapped out, so the thermostat shut off, and it was a whopping like 67 degrees in the room, and it was dropping down to 60-ish at night. And so then I got a new probe, hooked up the thermostat, waited 2 weeks, fed the subox. They both ate, they both digested, they both made perfectly fine poos. But in the meantime, I have antigen tests here. So I did test the regurgitated mice just to be safe. But because I understand the disease, because I scienced the hell out of this thing, I knew, okay, it may not actually be crypto. It might just be the temperature. And that gave me a peace of mind, and it kind of dropped the anxiety, and it made it so that I could make it through and make an informed decision. I think one thing that people need to really do, and this is where this podcast is great, is do the nerdy deep dive and learn about the biology of things beyond just your snakes. And for that matter, now on my soapbox, learn the biology of your snakes too. I had somebody, I asked one of my students who loves snakes where they had a ball python. I was like, well, where do they come from? And he's like, I don't know, Arizona. It's just like, holy hell.
Natural history is the best care sheet.
Oh my God.
How's that person going to know anything about ball python biology? They think they freaking came from Arizona. So I think that also just educating yourself beyond the paint job and the enclosure, and just know a little bit of biology, it goes a long, long, long way.
My guess would have been Florida.
There you go.
Well, and in that same vein, I would say that Steve and Zac and I, being very much academically immersed, we know the proper search strings to look these things up so that we can deep dive.
You also have venues to do so.
Well, we do
talk about that because you do too, believe it or not.
Yeah, you do. There are ways to get past paywalls and things like that.
That wasn't a jab. It wasn't.
No, I know.
No, it wasn't interpreted that way.
It's a valid concern.
Yes.
Some people hit a paywall and then they just give up, not knowing that there are ways to get around those paywalls. But again, from the educations that we've had and the way we have immersed ourselves in this culture, Zac and Steve and I know, like, there are some ways to search for things that net you better results than just typing in how kill crypto.
Right.
Google isn't always the best answer. There are some databases. So if you're trying to find information and you're struggling, reach out to people who can help you. You know, Zach, myself, Steve, Julander, you know, all of us were happy to help you. You know, we're not going to spoon feed it all to you, but we can point you in a direction so that you're better able to find the things you're looking for. You know, one place I very strongly recommend, rather than going to Google and trying to find everything, you know, go into Google Scholar, because that's going to take you looking for actual scientific papers. If you want to take it up a level, go to PubMed. You know, that's literally a giant database of scientific papers. Now, again, you are probably going to hit a paywall on a lot of them, but there are ways around that.
Yeah. And even if you are apprehensive to pay, because don't get me wrong, I paid for plenty of papers because I wanted to really know. And what are they, $30, something like that? It depends on the paper, obviously, but even just reading the abstract, because the abstract's always free, you know, and at least that gives you an idea of if you want to pursue that paper more, you know.
A lot of them were on Wrinkles, weren't they?
Actually, 99% of the Wrinkles ones are completely free because they're all old as dirt. So I have like 25 Wrinkles papers I need to catch up on.
Can
Sorry,
you
go on.
post the question that Jessica just threw up?
Yeah,
Scroll up
it was back a little bit.
to dig through all of the hecklers' comments.
This one?
Yes.
Does anyone want to comment on bovine serum treatment for symptomatic crypto-infected snakes? There's some info in the literature, but just case reports.
I can talk a little bit about this. I talked to a lot of zoo vets about crypto when I was doing the deep dive, because a lot of colonies of some of the more endangered colubrids that are in zoos have crypto pop up in them. And so the zoo communities have to really answer that kind of key question when it comes to crypto, which is like, are we going to burn the house down? Well, we can't burn the house down because there's only 12 of these snakes left on planet Earth. So what are we going to do? And so they've really kind of tackled the, can we get rid of it? And my understanding in, 1030, so this isn't, I'm not at my prime right now, but is that the horse serum, the bovine serum strategy for dealing with crypto and reptiles doesn't really work at all. Right now, the main drug or main treatment that's being tested, and there was a paper that just came out on indigo snakes, like within the past 6 months, is Paramamyasin, which is an antibiotic that the Bronx Zoo used successfully to beat back a crypto infection in a king cobra that was imported that they ended up with. We use that at West Liberty. Here again, please do not interpret what I'm saying as gospel. So not like we all need to go buy Paramamyasin. We have evidence. We're not concluding yet because the timeline isn't long enough. But we had crypto pop in some Barons racers and in some black milk snakes, and the snakes were perfectly fine. The only reason why we knew that they had crypto was we did the tests, and the tests said they had it. Like, they weren't, no regurgitation, no stomach swelling, no liquefied defecation. And we treated those snakes with Paramamyasin per the paper that the Bronx Zoo put out on. We used the same dosage as them. And we ran our PCRs and did all that jazz and basically had crypto in the feces, and then we didn't have crypto in the feces. But remember, that does not mean they don't have crypto, and that does not mean that Pero killed it off. That means when we took the swabs in the poo, the spore count was so low that it didn't present itself. So I can't say that it eliminated it. That seems to be a drug that could work. But the indigo snake paper that just came out, they did the exact same thing. And they basically said they did not have evidence that Paramamyasin was able to knock back crypto. So the way I interpret that is that there are well over 1,000 colubroid snakes. So it just makes biological sense that Paramamyasin might work better in one species than it works in another. It could also be that the strain of crypto that was in our Baroni was extremely lethal to corn snakes, was just kind of bad to a Barons racer. And since it wasn't in its preferred environment, i.e. in the right host, when we hit our animals with paramamyasin, it knocked that particular haplotype or strain of crypto out. But if we had a Baroni, a strain of crypto that really does well in Barron's racers, the paramamyasin may not have knocked it out at all. So with crypto, the infamous cure, we don't know what it is yet. We just have evidence right now that paramamyasin might be a drug that works on occasion. That's the way I would word that.
It's something to look forward to in further testing and so on.
But to kind of drive this point home about we don't know that much about crypto. So the first time we isolated the cryptosporidium from our snakes, I describe and study crayfish and do taxonomy. So I understand how this stuff works. Yeah. And so we sent the samples off to get sequenced, and it came back as Cryptosporidium spa. And I was like, what the hell? And it wasn't Cryptosporidium spa. It was Cryptosporidium spa 1, Cryptosporidium spa 2, Cryptosporidium spa 3. So, and then Cryptosporidium serpentis.
Wow.
So
to clarify here for people who are not familiar with the jargon, SPA is short for species.
Yeah, it means it's undescribed. So, we could have found 3 undescribed species of crypto in our freaking corn snakes. Or, it may not be. So, that's the thing when it comes to the science of understanding this snake disease and all that kind of stuff, everybody needs to understand that, we haven't even put our foot in the water yet. Our toe tips are in the water, but our foot isn't immersed yet. That's how little is known. That's why Steve doing the research he's doing is so incredible, because he's answering applied basic questions, but the word basic here is not bad. Basic here is foundational.
I would guess that Steve would say the same thing is seen with the serpentaviruses. The test can show that you have a serpentavirus, but you're going to need to do some kind of molecular level sequencing to necessarily say if it's, serpentavirus from this cluster or from this cluster or from this,
Right.
And they do, if you look at the papers or if you talk with Steve Moore, they do form little familial clusters, and each cluster behaves different from each other cluster. So if you just test and get Nido, it could be bad cluster, it could be neutral cluster. Mm-hmm. You don't know.
I will say, there's one cladovirus that we found in 2 species, and out of over 60 positives, it might even be as hard as 80. I've never seen a single animal with any clinical signs from that specific clade.
Okay.
Yeah, because I think you would mention corns and their version of it. And
there's those, some colubrid viruses.
At the last Carpet Fest, you had talked about it and how it's there in corns, but it seems to have 0 effects on them, right?
I'm actually not sure about that one. I don't know. Yeah, I don't remember. The prevalence in colubrids is just really the prevalence in most stuff outside of pythons is just very, very low. So when you do find something, it's either you got lucky or, it's part of testing a wider colony and they happen to have, something else and that happened to be picked up. But there are probably at least, I don't know, half a dozen different clades at least in serpentavirus right now. And nestled within that is a cow nidovirus and, a couple of fish viruses and I think an insect, it's not like this is the branch that infects snakes. It is, this is the branch that we're just starting to open up a little bit of understanding for, and we're finding them most frequently in python.
And real quick before we keep going, Scott Ipers in the group chat, and he had mentioned that ResearchGate, which is one that I frequently use. ResearchGate is a bit of a handful if you're not in academics. You basically have to apply to them, and if they think that you're worthy or whatever, they'll give you access. But definitely check out ResearchGate. And then Scott had also mentioned that in all the years that he's been not necessarily in academics directly, he's never had an author say no to him asking for a paper.
Yeah.
So
Yeah.
If you have a paper name and copy and paste it into normal Google and just put file type PDF, like
Yep.
4 out of 5 times that you'll find that paper.
Yeah.
Yeah.
It's like
these are some of the tricks. But yeah,
knowledge.
Scott's right. If you just email the lead, the corresponding author, and when you look up a paper, there's little notations next to all of the authors, and one of those will say corresponding author, and somewhere in there it will also have the email address for that person.
Yeah.
Now, if it's like a 40-year-old paper, you might have to work a little bit harder, but just email them. They will have a copy of their own paper, and they will love to share it with you.
It's the paywall for the sites that are hosting it,
right?
Like the authors aren't getting any of that, right? Like
No.
just for the website.
Yeah,
okay.
that's
the
for the periodicals that
the
are publishing.
servers and stuff like that. Yeah,
the authors want to give you their paper because they want to share that information. I mean,
Right.
We as scientists don't do this just because we get our giggles writing papers that make us tear our hair out, that we want to shoot ourselves after reviewer number 3 comes in and picks everything apart, and then we have to rewrite the entire paper that we spent a year writing. That's not fun for us. We do it because we like doing the science, but we also like sharing what we have learned.
Yeah.
I mean, that's kind of the point of doing the whole study, isn't it?
Yeah.
Yes.
Yeah.
No, people reach out to the author. Now, granted, if the author is some powerhouse author that has a giant lab and is writing grant after grant after grant and is in the field all the time, that's one thing. But most of the time, that's not the case, and I can attest. I like it when somebody. This is what happens when I get the email of somebody asking me for a crayfish paper. I literally open the email and I'm like, "There's someone other than me." Oh yeah. Okay,
Someone
cool.
else wants to read about mini lobsters.
Yeah, and then we send out the paper about the mini lobster. Like, there are douchebags out there that will not give you the time of day, and that's where people like us get this reputation in certain circles for like pinkies out, ivory tower, dickhead academics. But that's the people that are here are not those people, and they don't ever want to be, and so reach out. And I've gotten papers for people. I've had people ask me, I'm really interested in whatever, can you help me? And it's enjoyable for me to do a search for these things. Now, granted, I'm not advertising to the universe right now that I will get all your papers. But at the same time, learning the tricks is kind of important. And ResearchGate's a great one. One of the things I make my students do the second they enter my lab is that all of them have to get a ResearchGate account. Because I'm at a little school and we don't have big, powerful libraries. So I'm in the same boat most of the people listening to this are in trying to get the literature. So, it like Steve said, go to Google Scholar, not Google, and then type in your key phrase, then add PDF somewhere in your search. And a lot of times, not a lot, if it's a more recent article, you can get around it. What will happen is it'll actually take you to the ResearchGate page, and then you can download it.
So maybe it's a dumb question again. With both crypto and serpentoviruses, isn't it counterproductive to have a mass die off? Like, is that like what? First of all, what ecological niche does crypto even fill? Like, why is it even there?
It's a great question.
Like, what's the point? It's like sand gnats. Like, I cannot fathom why sand gnats exist. Like, you're too small for anything to really eat. Like, you're just there to be annoying.
It fills its own niche.
It
fills its own nipper.
So what's crazy about parasites, and you can go down massive rabbit holes with parasites and nerd out hard, is that a parasite's ecosystem is not necessarily the ecosystem you're thinking of. So you were thinking of ecosystem maybe as like forest, and then the forest floor, and then the stream running through the forest. And the way a parasite looks at an ecosystem is its host is the ecosystem.
Right.
So basically, a corn snake is an ecosystem to a cryptospore. And so, or an individual cryptosporidium. And so when it gets inside the snake, there's different ecological communities in the snake. There's the snake's lungs, there's the snake's bladder, there's the snake's liver, and the gastrointestinal tract. So there's complete niche partitioning of the actual snake's body when it comes to the crypto. And so with parasites, you're absolutely right. The goal is not to burn your house down. The goal, though, is to reside in this organism and take enough of its energy that you can survive, but not so much energy that you're going to kill it. And so that's why asymptomatic carrying in Cryptosporidium is probably what the organism wants. They're going to get, when they get to the point where like the snake is spray painting all over the forest, that's like, oh crap, we got to like bail out of the ship. The snake's going down.
Mm-hmm.
And so that's why when you kind of look at parasites through a biological lens, snakes can live with them if they reach a homeostatic state because they're living with them all the time. Like if you actually do a necropsy of a dead snake on like it from the wild and you look in all the different there's freaking parasites everywhere. They're infested.
Lungs,
and
liver, kidney,
brain,
everything.
Kidneys. But all those parasites have reached an equilibrium, just like organisms reach an equilibrium when they're out in an ecological community. It's when and so it's kind of a really weird thing to think about, but that's what's going on. So when you bring a snake into human care, that's not natural. And so we are putting our perceived idea of what constitutes the environment that this thing needs to live in. And there's things going on that we don't think about that could then trigger these, the snake's immune system to start to tank. And then you see the expression of the pathology and all's well and good. Like one thing that I am coming to a realization here in Northern West Virginia that I didn't really think about until this year is when the heaters kick on up here in the Great White North, our humidity just plummets.
Mm-hmm.
And when you go from a very humid environment to no humidity in a matter of 2 weeks, that is going to cause physiological stress. And so these animals that evolved like in South Carolina, where you have relative humidity all year long, you bring those Okaticorns up here, and now some of them aren't going to get stressed. Some of them get stressed to hell and back. And then we end up getting the crypto popping. I don't know if humidity is what's doing it, but thinking about your collection that way is what could kind of help you get out ahead of these potential outbreaks. So next year, I'm going to try to do something. Not that I had a crypto outbreak, because I did, but I just want to make the ease as easy as possible.
Sure.
I'm actually going to be focusing a lot more on my humidity because I got govies and I was actually able to see.
Those things are fun, man.
Oh my God. Nerd time.
Yeah. You never get so excited to refresh a little graph and see what things are looking like over a night.
Yeah, but just in a 24-hour period of time, my humidity is going from 80% to 10%. That does not happen normally in nature. Yeah. So
and that's a good, that plays into the the pythons and the viral aspects as well. Is think about how you as a person feel when your heater in your house starts kicking on, dries out your sinuses, you're waking up every morning and your throat is sore because you're getting all dried out and stuff, so you hear your snakes wheezing in those first couple of weeks that your heater is kicking on. Maybe your snake isn't wheezing because it's actually sick. Maybe your snake is wheezing for the same reason that you're waking up with a dry throat every morning, because your house is just all dried up and you're drying them up. So, if that's stressing you out, obviously that's stressing your snake out too.
Yeah.
Get on top of that shit. Don't just be like, it's winter. They have the wheezes every winter.
Yeah. How many people, they get a nose.
That.
Yeah, exactly. How many people get a nosebleed every time they go in their basement in December? It's like, well, you ever wonder why? Well, that's why.
It's not just in my basement. Like every year at late winter and every year at early spring, basically the transition, I pretty much get a nosebleed every single day. It just my sinuses get all messed up and just I've had that happening, for decades. It's just the way I am.
Yeah.
To kind of build off of that as it relates to serpentivirus in terms of balancing with the host, I think that is what we see in the majority of cases, which is why we see the majority being, asymptomatic positives, oftentimes, unless you're hunting after a sick snake. But that's kind of, in my mind, why I'm a little bit more fearful of like a chronically sick snake in a colony. If you have a colony of 100 of the same species and you have all of them in the same conditions and one of them is sitting there with illness, in my mind, where I've placed that animal is, I don't know exactly the equation that resulted in that animal being sick, but somewhere in that equation is the parameters I have in my colony and whatever virus this snake has. So clearly there are circumstances where that virus can be potentially pathogenic. And so I will.
You're
Lost
still
your
there.
volume.
Bark twice if you're in Milwaukee.
It's like a bad Godzilla for a second there.
Oh, he froze.
Oh no, God, we lost him.
I think he's still there.
He's
just
still moving, but we lost his mic.
Is there any possibility, maybe more so in the viruses rather than crypto, that there's a potential for like total sort of, not necessarily immunity, but to where the hobby as a whole, like things have it, but it's not killing things off?
No, I mean, yes and no.
I'm on team no.
Well,
Travis knows more than me.
And well, generally, I do agree. The ultimate goal of any virus, of any parasite, is to reach, like Zac was saying, that
homeostasis,
that equilibrium where you're not torching everything down. Now, the flip side of that is there's this constant, evolutionary arms race, basically, where the virus is changing, the host is changing, the virus is changing, the host is changing, the virus is so
right.
You can get there eventually, but you're talking sort of not fully geologic time, but a lot of geologic time. If you look at us as a human species, so we have all most likely been exposed to mono. That's CMV, cytomegalovirus. In almost all cases, it's very benign, and a lot of people get it, never show any symptomology. They carry the virus for the rest of their life, and it does nothing. But even then, there are some people where it causes a huge problem. You get people who have just chronic mono. It comes back every year, every 2 years, every 3 years. The virus reactivates in the most horrible way in some people and becomes cancerous.
But is that taking advantage of a compromised immune system? Like that's just a byproduct of someone?
Sometimes yes, sometimes no.
Okay.
But again, so ultimately, that virus has hit, on a broad scale, we would say it has hit sort of an equilibrium with us where it is benign. But in an absolute sense, it's not completely benign because there are still some people who will always have some level of susceptibility. And in some respects, I think we can kind of see that with these nidos. Like, we have those ones that Steve has seen where, he said he's got all of the ones that he's found and they've never seen a case where it has caused active infection. Now, that doesn't mean that clade will never, ever cause an active infection. Tomorrow, Steve may find the one animal that gets sick from a virus in that clade. Or Steve may find another 7,000 cases, and 30 years from now, the one that causes an infection in that clade will pop up. So you can, over a certain amount of time, reach this equilibrium, but it doesn't happen.
It's not a short-term thing.
Right. Yeah. It's not a short-term thing. And if you try to make it a short-term thing, usually what you see is like is the mass die-offs. You know, if you're just exposing everything and searching for those, you know, 1 or 2 animals that aren't going to be infected, and then their offspring that also aren't going to suffer, you know, you're gonna burn through animals like this. And I really don't advocate for, let's just burn the house down by getting everything infected and force evolution. You know, it's not a good, safe way to do it.
Steve, my question before you dropped out was, is there ever the possibility of serpentovirus, be it in a single species or sort of across the board, ever just becoming to a point where it's just, it doesn't wipe out anything? It's everyone's got it, but it doesn't do anything. It's just completely benign. It's just there.
Can you hear us, Steve?
Oh, sorry. I'm like, my audio seems to be cutting out. I'm only hearing about every other word.
I asked, I don't know how to do it without it doing it again. Basically, like in serpentovirus, be it a single species or like across the board, I asked if there was ever a point where it would just exist within, you know, the population being the hobby, where everyone's got it, but it's not killing anything. It's just there, it just exists. It's
Sorry, I like can't hear anything you're saying, but I can potentially jump back into where I was before I got booted off in terms of, you know, those kind of slow burn snakes in a colony. You know, when you have this animal where there are circumstances where disease can be present, and that animal is just sitting there with that disease, you know, if it is sitting there mutating virus at 10 times the viral load, that equates to 10 times the mutation, which really could present a laboratory to come up with something a little bit more virulent. So that is where I get a bit more paranoid about the animals that actually develop clinical signs in an otherwise healthy colony and that don't get better, but maybe not, you know, progress towards in and of itself a fatal illness. Yeah, it's just that sitting there being sick is not good.
And then Bruce, what is that? Shilling Law. He said, "What effect would bioactive or more natural housing have on these diseases versus more traditional methods of keeping?"
Do you want to go with viruses first?
Well, I mean, I already think I know your answer, Zach. Is it gives the spores a lot more places to hide and makes it harder to clean. So
just giving them a mansion.
Yeah. However, I still don't think it. So you're getting into welfare now, that dirty W word.
Ugh.
But it's an important word that I don't think should be all that dirty in herpetoculture anymore. Because if you're keeping your snakes completely sterile because you think they have a disease, you could, in theory, possibly be inducing stress because they don't have the security that they might get from a more naturalistic setup. And I'm not talking about all snakes, but at the same time, every snake that has been studied so far, when you give it more options in an enclosure, it seems to do better. So if we're saying that a non-stressed snake is not going to display disease pathology, then we're kind of getting into this weird way of thinking where if we provide them more options and they feel secure, then they're not going to be stressed. Their corticosterone levels are going to be low. And if they're not stressed, then if they do get one of these diseases, it might help keep the disease at bay because they're feeling secure and, you know, content, I guess would be the word that we're looking for.
Yeah.
If you are keeping the snake in a sterile environment, sure, you're going to be able to get the spores and clean all that kind of jazz. But if the animal's permanently stressed, you're also creating an environment for the pathogen to just basically go hog wild. So there is, I hear a lot of people kind of talk down on naturalistic keeping and bioactive and all that kind of stuff when it comes to the management of diseases. And there's definitely an aspect that complicates things. I'm not saying that. But you got to understand that, like, maybe because the snakes or lizards, whatever it might be, are more content, and that's going to keep the disease at bay. Now, the extreme opposite end of this is that, well, you know, Loughman just said then that Bioactive creates the best situation on Earth, and therefore it's the best way to go because there's people on the extreme end of that continuum as well. And I'm not saying that because there are certain situations where, once again, we anthropomorphize and we basically create an environment that we think the snake really wants, and in reality, it may not be what it wants at all. I've got snakes right now in racks behind me that I've tried to keep in a naturalistic setup. They will not eat. They get stressed to hell and back. I put them in the rack, give them 2 inches of substrate, and happy as larks, or content, whatever the right word is, and then they start putting on weight and everything's good. So, like, I'm not saying Bioactive is the way per se. What I'm saying is giving the snakes choices and giving them the ability to go where they want to be. And that's what could be beneficial, and that's what naturalistic keeping. That's the goal of it. So that's kind of a more complicated issue than one might think, in my opinion.
I think you summarized it exquisitely. Honestly,
My audio seems to be working fine, so let me know if there's any questions that I'd missed in that.
Do you want to go back and try the one that I was talking about before?
Yeah.
So, okay, so
definitely got to recap that.
I had asked, and Wyman sort of touched on a little bit, but as far as serpentovirus, be it in a single species or sort of across the board, is there ever going to be a point where there's sort of quote unquote herd immunity and it's in collections, but no one's having any issues with it? Or is it just so diverse and so large and constantly evolving that it'll never be able to fully be pinned down?
Yeah, it's just too big. Yeah. Yeah. For the same reason, I wouldn't set expectations of a vaccine or anything like that. It's just, there's just, we're talking about it like
about that too.
Yeah, we're talking about it like it's one thing, but we're talking about, conservatively 50 viruses when we're,
Well,
and is that a growing number too? Is that?
Oh yeah.
I mean, think of it like, in some respects, think of it like flu. There are a bunch of different flu types, and every year we're having to get vaccinated against the flu, and those vaccines are not 100% effective when we get the flu vaccines. And that's a virus that impacts us, so it kind of matters that we get vaccinated against it and there's a vaccine against it. In the big picture here, there isn't the funding or the interest by the world at large to invest in a vaccine for these snakes. I mean, we as reptile keepers like to think that snakes are really important, and to us they are, but to the world in general, nobody cares. Nobody is going to waste time, money, resources into making a vaccine for snakes. It's just not going to happen. In fact,
it's not a health crisis.
Yeah, forget the vaccine.
Yeah, it's not. It's forget the vaccine. It's look,
studying them in general. Jeez.
Right. I hate to sound like I'm shitting on the hobby, but in some respects, I kind of am. Like, we have all of these auctions for USARK, and don't get me wrong, USARK does great things for us, and people need to fund USARK better. But like, people dump stupid money into that. Like $5,000 for a pencil, and yeah, it's great that we're donating. But then, like, when we have fundraisers for Nido, it's like pulling teeth.
Yeah.
But then the hullabaloo that comes up when we're talking about testing for Nido, it's like, well, if you people really cared and you really wanted answers, maybe you should be putting some of this same enthusiasm into donating to the auctions for Nido that you put into donating for the auctions to USARK, because that money can then be used to better study these things, to better understand them, and then we're a lot less likely to have these, flash in a pan panic attacks that we saw last week, or that we see when somebody comes out because their entire collection melts down.
Yeah.
And like I said, I know it sounds like I'm kind of shitting on the hobby, and in some respects I kind of am, but don't sit here and say, well, we'll just develop a vaccine, because that's so far from possible.
Yeah, I think it was just an idea that someone may have had, but and I don't think you're shitting on the hobby at all.
I know people have asked before.
Yeah, I don't think you're shitting on the hobby at all. I think that you're saying something that the majority of us in this, at least our collective circle, have thought of and have expressed to themselves, and needs to be spoken up more about. You know,
I mean, it's an unpopular opinion, and
sure,
sometimes I just cut straight. Sometimes I just cut straight to the point, and I'm not going to sugarcoat it. It's just what it is. And like I said, great. I'm not trashing USARK. I am not trashing the USARK auctions. I find it bothersome sometimes that as much as people say they care about this industry, there are places where it's important and they just kind of they act like it's not. And that is very frustrating to me. And this would be one of those. If people donated more and contributed more, we could do more research into these things. And not just for serpentovirus, for adeno, for arenaviruses, for paramyxoviruses, for crypto. We could get more information out there for everybody.
Yeah, there's ones we don't know about. Like regularly, we get weird cases in the diagnostic lab, and it's not any of the common things. And then we'll sequence it, anything in that sample, and come up with some weird virus that we've never seen, or bacterium. There's herb medicine is very much a fluid thing that is ever improving, I guess.
Well, it doesn't seem to be exactly on the cutting edge compared to,
that's correct.
Other commonly household kept pets, it's
sure. Well, it's still an up and coming pet, for lack of a better word.
Yeah.
You know what I mean? We have more people in herpetoculture and herpetofauna than the history of humanity. And it's going to take time for medicine and science in the captive world to catch up, So,
yeah.
I also think that I don't mean to cut you off. I'm sorry, but there's also a stigma that comes from multiple generations of herpers back, where it's the vet doesn't know anything. I know my animal. I'll just do X, Y, and Z. Or, there's nothing you can do. Or my favorite quote that people have heard me say many times is, those things die great. They die really well. So there are people of all different generations that still hold on to, myself included, an attitude like that. And now is the time that we're seeing all this stuff with all these diseases and illnesses and the ability to have the knowledge in literally the palm of our hand. The progress, we just need to keep the progress going and not fall back into those old ways. You know what I mean?
Yeah. Yep. There's many challenges in conversations regarding this, because, you can have intellectual conversations about pathogens themselves and how they deal with hosts and stuff, but where it really gets tricky is how to convert that into policy that people within the industry can actually operate by, and that's fair to everyone. Mm-hmm. I mean, yeah, and I guess bringing this back to the beginning in terms of what Morph Market suggested in terms of dealing with serpentoviruses or nidoviruses, I think how I would do it is, I know if I test a snake here, it'll test negative. Whether or not it's positive, it's going to test negative, because it's very happy. It's a very happy snake, and I am very aware of the strains that I see in my colony, and I know that the strain I have, I can't detect unless there's a reason to detect it. Mm-hmm. So from a seller's point of view, it makes way more sense to test on the buyer's end after that animal has the stress of shipping, and where you could have that little, it's going to give you your best chance at a positive sample. So in terms of dealing with the real world, I would say a negative test on or shortly after arrival is pretty much all any seller can reasonably guarantee.
Well, I guess that kind of ties into the issue that we saw recently with carpets and the whole thing fiasco with like star pythons and tests a year later coming back positive. Then at what point is it, if someone just tests and the first test comes back negative and then they test again a month later, 2 months later, 3 months later, and that goes back positive, how do we know that didn't initially come from that seller?
Yeah, well, that goes to what Travis was saying the other day about sneakers, right? Was that yesterday we were talking about sneakers? you buy a pair of sneakers and you take them home and you try them on, and hey, they don't fit my feet right. I don't like them. And you take them back the next day, and the store gives you a credit or a refund or whatever. But if you wear those sneakers for 6 to 9 months and they're worn out and they're dirty and they're tattered, and you say, hey, these don't fit my feet right. I wore them for 6 months, but they're not right for me. The store's not going to take them back, and the store's not going to want to deal with it because you already quote unquote used it. So
I'm just talking about from the standpoint of like the origins of it,
right? And that's what I'm getting at. It's
the same thing. Like you could have this Nido that's sitting silently in your collection.
But what I was saying is that it could have come from something else.
Right.
Or it could have come from the collection in the first place.
It could be in your collection in the first place. High. And when you tested the snake when you first got it and it was negative, it was legitimately negative. But then, 6 months in, as you're getting ready for breeding season and you start pairing up your animals, and now this animal gets a little bit of a wheeze to it. And like I said, is it the wheeze because it's actually sick, or is it the wheeze because your heaters have kicked on and all of your animals are getting dried out and their lungs are getting a little scrapey, so it's wheezing, and you test because you see this symptomology and now it tests hot? Well, is it testing hot because you've got the Nido floating through your collection? It's a benign one in your collection. Your animal in question was clean when it came in, but it got it because the rest of your collection has been circulating it. And what you're calling the symptomology isn't really the symptomology, the same way, Zac had the Subox yak up and first thought, oh God, crypto. But then he stopped and went, well, no, wait, maybe not crypto, So you have to take it all into effect. And, I said, that's kind of what I think John was trying to get at a bit with the policy that he did was, saying that you have to let people know if you get a sick snake in 4 hours doesn't work necessarily with the testing, but he still put a limit on, if you're going to test, it has to be within 2 hours of receipt, and you have to do video or photographic evidence of that within 2 hours of receipt. Because John was well aware of, we at Morph Market don't want to say 6 months later, 10 months later, I tested this animal, it was positive. But if you as a buyer want to test the animal as soon as it gets in, you should have that right. The same way if you buy a cat or a dog, you should have that right to take the dog or cat in for a wellness visit really quick off. And, I think both sides lost sight of really what John was trying to get at, which in my heart, I think was a very legitimate and legitimizing way to make the hobby go. But you had one half of the hobby that was like, you're telling me I have to test, and that's not cool. And if I say I'm not going to test my animals, now you're going to say that I'm uncaring and I'm money hungry. And the other side saying, you can't tell me that, I can't test because you're a big breeder and you don't believe in this stuff, and that I'm just paranoid and, I'm out to destroy all the breeders. It's not, if I want to test an animal, I'm going to test an animal.
Right.
I don't care if the breeder tells me I can test the animal or not. Now, if I test an animal because I chose to, and it comes hot and I talk to the breeder, I hope the breeder's going to make it right. The same way, if I buy a dog and I go and take a wellness vet, visit, and it comes back sick, the breeder's going to make it right then. Now, does the breeder have to? No, but the dog breeder doesn't have to either. If the breeder doesn't, should I blast them publicly here, there, and everywhere? No, I shouldn't. Am I going to be upset if they don't make it right? Yeah, I am. Am I probably never going to buy from them again? Very probably not. But, again, this hobby has a tendency where something goes wrong, and then the next thing we do is just broadcast to hell and gone about how horrible somebody is. And, again, I think both sides lost sight and spent too much time nitpicking at each other than to realize overall what was asked wasn't terribly unreasonable.
Yeah.
And ultimately it would have been legitimizing. And, again, this is me personally. I'm not talking for John. Again, at the same time, I think that the conversation is out there now. And I think people will have to consider it more. And I hope that, at least discussing what we've been discussing here, talking about, these things are prevalent. These things are probably common. There's probably a time and a place to test. There's probably a time and a place to panic, but it's not every single time, and it's not all of the time. And I'm hoping that both sides will be able to step back from the edge a little bit and kind of focus on, working to make this right for everybody.
Yeah.
And that's my hope. And like I said, maybe it's a pipe dream, but yeah, rationality is what I'm looking for.
Yeah.
I mean, the way I see it, the solution seems extremely easy. It's either you buy from people that test, or you don't buy a ton of animals.
Or you make the decisions as you need to make them.
Right.
Like if you're shopping for animals and you come across someone and they say they just don't test, if you don't like that, then don't buy from them.
Exactly.
Right.
Or again, this is, you can mitigate risk by, an animal that you yank out of an egg and then stick in your collection is probably a safer bet than buying a 5-year proven whatever from some breeder. Not all animals are necessarily representing the same amount of risk either.
Right.
True.
Well,
and there's a difference between
animal.
buying that 1 5-year breeder animal and buying 15 or 50 snakes because somebody's decided they're getting out of the business.
Yeah.
You have to weigh the risks of what it is you're doing.
And that's sort of been my stance with all of it, with my collection, is like, just be smart, like pay attention to possible cross-contamination, pay attention to, how things are being worked, like just be aware of it, be aware of what you're doing, be aware of where you're putting things, be aware of disinfecting, like be aware of just pay attention.
I think it also comes down to, we talk about how this is an industry, this is a trade, this is a hobby, when we all know this is also a community. And I think a lot of times it's a community above everything else. And I don't know about you guys, but yeah, have I bought animals from breeders that I didn't know personally? Of course, most of us have. But if I have the opportunity, or I see a window where I can maybe communicate with them and try and be friendly and get to know them and make it personable, then I'm going to do that as well, because I want to know how they took care of the animals, how their breeding is, lineage, all that stuff. So going back to what Justin was saying is, if the person doesn't want to test, or you don't like their practices, no, you don't have to buy from them. But at the same time, ask them, talk to them, get their opinion. You know what I mean? And if they're a total jerk and want to shut you out, well then, there's plenty of fish in the sea, so to speak.
There's very few scenarios where you won't be able to find that same morph or animal.
Right.
Like it's not the last one on the planet. That's what I tell myself all the time to talk myself out of buying things. It's like, that's not the last one to exist.
Yeah.
And I mean, that engagement thing I think is important. So I mean, I hatched those kukris this year, and when I put them up, I got a lot of interest in them. And I had one guy who, he just blew me up with questions. And every single time he asked me a question, he always apologized for asking the question. And every single time I told him, I was like, stop apologizing. I much prefer that you ask these questions, because if you're asking these questions, I know that you're actually caring about, picking up these animals and what it means to take care of them.
Yeah.
You're doing the research. You're getting yourself ready and prepared to really take care of these animals. You're not just like, ooh, pretty snake, I want it. Now, at the same time, I did have one guy who literally said, give me your PayPal now, I want. And, I gave him everything that he wanted to. But, afterwards, I found out that he'd already worked with kukris and he was looking for new bloodlines. And that's why he wanted my animals. Yeah. But he came back to talk to me later. He was afraid they were going to disappear. So that's why he was just like, PayPal now.
Yeah.
But this other guy who, we went back and forth for like 4 days asking me questions so that he could be, comfortable buying these animals and knowing that he was going into it with animals that he knew he could care for.
Yeah.
And I'm happy that he did that. And I was glad to answer his questions.
Yeah. I think a lot of us in our group of friends, as well as friends of friends and cliques of cliques, right? There's there's always one or two individuals that are just like us who are sick and tired of nonsense, especially when selling offspring. And what happens is, I don't want to say they're jaded or ruined or whatever, but you know, you get these messages that says, you know, do you have gecko for sale? Okay, well, are they not from the United? Well, not even that. Do you have gecko for sale? And it's like, okay, are they just bad at typing? Are they not, you know, American? Is the English not their first language? Or do I just assume they're an idiot? Are they a minor? Right. Or do I just assume that they're an idiot and I treat them as such? No, I do a little digging. I'll look at their page, whatever, you know, and then I get another guy who I don't sell anything. I don't breed anything. I really don't have anything for sale, but I get a guy who messages me on Facebook. I have no idea who this gentleman is, but he messaged me. He says, hey, somebody told me that you work with Cordylidae. I have Tropidosternum. Would you be, you know, able to look at a couple of pictures of their bellies and tell me if my gender ratio is correct or not? Him and I talked, just like, you know, Travis was saying, for like 4 days, sending crotch pictures of lizards back and forth. So like, and I could have said, hey man, go out and buy, you know, Jans Riesing's book and flip to page 412 and look at the pictures. But no, we talk and we socialize because it's a part of this community, you know, and people also will totally understand if at that moment in time you don't feel like talking and say, hey, I'm swamped, I'll message you tomorrow or whatever. And if they don't message you, you can't get mad. It's life, you know. So I think the perception of how right. Well, no, it's the perception is how we deal with things in our community. We can be pissed off at the nonsense, or we can overlook the nonsense and focus on, you know, the Chris Panchab glass always full, you know. So
he's a freak.
He's beautiful.
I love him to death, but I don't know. I'm not like a negative person, but I'm not nearly as positive as Chris.
Right.
Sometimes I think he's just on some sort of stimulant or drug.
But Steve, you were going to say something before we got into the whole buyer seller convo. I feel like you were going to chime in and we stampeded you on accident.
Oh, I don't remember what it was. Yeah.
Okay. Oh, you'll remember at 2 a.m. and text us.
No, I'm sure. Yeah.
I was talking to Jessica a couple days ago, you know, through messages and stuff. She had dealt with arenavirus. I'm very unfamiliar with that. Is there any
IBD? Yeah.
IBD.
Oh, okay. Okay.
Yeah. So IBD is Reptorrhinavirus. You know, everybody knows it as IBD. They don't typically associate it with its more scientifically known name.
Gotcha. So what should be people's sort of best practices, like your top 3? This is how you can make your life a lot easier and avoid heartbreak.
For crypto, I think it depends on the number of snakes you have. So if you know going into this that you don't aspire to have a giant collect more than 20, and you're just really, really selective, I don't think it's beyond anybody's. I think it's a totally fine request especially if you're buying the snakes online or you're reaching out to people, to ask before you buy the snake, hey, what happens if I get a positive crypto test? And just ask the question, point blank, and deal with it. And if you do that, then I think that at that number of animals, it is possible, if you're vigilant and you practice good quarantine and you know what the symptoms are and you test yourself and blah, 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 that you can potentially not have crypto in your collection. If you have a collection of colubrids and it's over 50 snakes and they're coming in, you know, from all over the country, or you're a large breeder, it's just based off every bit of evidence I have to date. It is disingenuous for you to think that you are crypto free, so you should act like you have it. And I don't think it's outside the realm of good practice to basically have your collection tested during those stressful times a year, like what Steven was saying. Mm-hmm. If you test in the middle of the summer when everything's hunky dory, you're not going to get a positive. But if you test when the animals are stressed, that's hard to say. coming out of brumation, going into brumation, you're gonna have a bigger bang for your buck. And I think it is within the realm of possibility for people to test entire collections and then have the crypto ward situation. You know, where you just basically move the animals off site, and then it's up to you as the maintainer of that collection to figure out what you're going to do with those animals.
Mm-hmm.
That's what I think is best practice. That's what we're doing here. That's what we're doing at the university. So, like as an example, I knew we were putting things down for brumation. I knew that the furnaces were coming on, which is like the great stressor of Arnett Hall, where all the snakes are at West Liberty. And so, one of my co-professors at West Lib that does the zoo size stuff with me, we tested the entire collection for crypto. We had crypto there before. We had animals that were stressed. I was totally expecting to get positives, and we sent off 80 crypto tests and nothing came back positive. And my response to that as a scientist was not yes, it was bullshit. Literally, like, no, sorry, don't believe it. So we're cautiously optimistic, but I'm not in any way, shape, or form convinced. So when everybody comes out in the spring, we have a large collection of snakes. I've got little undergrads feeding these things. They're learning sterile techniques. So we're going to test everybody again. And was it pricey? Yeah. And we're not making any money off of our collection. Our collection's purpose is to teach. But if you're making money off these things, I think it's just a good practice to do this. And then if you get a crypto positive snake, don't like that snake goes away. It doesn't necessarily get bred. You want to interact with it as little as possible. And then that just becomes the standard practice. That's based off everything I've read, everything we've done, dealing with crypto, the way we've dealt with it. That would be what I would do. And I'm testing my animals here as well because I have to.
Yeah, I'd rather test before I even sent one out because I mean, you think about that, like you're looking at the cost of shipping and then cost of shipping it back.
The
on top
problem
of the test.
Getting what Steve said is if you test before you ship,
oh right,
they're not stressed. If you test after they ship, they're stressed.
Yeah.
That's when you're going to get the positive in an asymptomatic carrier, in my opinion. Or your probability of getting a positive is higher in an asymptomatic carrier post-shipment.
Yeah.
Yes.
Yeah, I would say if I were to distill down my advice to 3 main points, it would just be, first of all, don't panic. These viruses are out there. Oftentimes they don't seem to matter, except for when they do. But, maybe wait for your panic until you actually do have a reason to think about those viruses really mattering.
Yeah.
With that in mind, prioritizing biosecurity about stuff coming in, have a really good quarantine. And I think buyer and seller conversations need to happen about what the expectations of a healthy animal are. But if I were to design a policy that would be fair to buyer and seller, it would look an awful lot like what Morph Market had drafted up. So,
well put.
Yeah, and it's hard for me to add anything really to what either Zach or Steve had said. Testing when and where it's appropriate, but you kind of decide when and where is appropriate for you based on your collection. Quarantine, and I will nitpick here slightly. 3 days is not a quarantine. 3 weeks is not a quarantine. I'm sorry. I don't care how important that male 17 gene animal is to get to your female that you just decided to pick up last month. That's really not a good quarantine. And I go extreme with my quarantines, but at the same time, I'm also bringing in some wild caught animals for some of the things that I do and working on establishing them. So, I have had animals in quarantine for a year. Again, that might be a little bit extreme for, something that's captive born and bred, but I don't think that 60 to 90 days is unreasonable, even with a captive bred animal, because you just don't know sometimes.
Yeah.
Watch your animals, know your animals. Don't automatically assume that everything equals worst case scenario. Sometimes a regurg is just a regurg. Sometimes a sniffle is just because it's that time of year where the heaters are drying everybody out. So use some common sense and don't always jump to the worst case scenario.
Well put. I'll ask the question that I'll ask a question no one wants to ask. This is completely an opinion of you gentlemen, Justin included, myself included. I want to know your guys' opinions. You have something that comes up positive. Things get isolated. Precautions get taken. Do you euthanize the animal or not?
Here we go.
Whoever, we'll start with Zach.
So,
if it is, I'm going to talk for crypto.
Sure.
If it is actively showing pathology, so we've got liquefied diarrhea, bloated stomach.
Yeah,
at
it's
that
past the
point
point of no return.
Of no return, it's being euthanized.
Yeah.
If it comes back it's asymptomatic, I'm not euthanizing it at that point. I'm going to ward it. I'm going to do the whole process I've talked about now, move it off.
Sure.
And then I'm going to really think about the ultimate. Why is that animal in the collection? Now, can I afford to have a positive animal here? What's the likelihood of it spreading? What's my fly count? There's literally, it is not just a quick knee jerk, burn it with fire.
Sure.
Kill it with fire. No, I'm really going to kind of think that thing through. And I've done this. For instance, right now in my closet, in my bedroom, there is a Boruna maculata, a musarana, that has popped positive for crypto 3 or 4 times. We've hit it with I've done the Paramamyasin. The snake is completely asymptomatic. I caught it just doing this kind of screening, and it's okay. I have a male Boruna at West Liberty that may be crypto positive and also asymptomatic. So in this scenario, the snakes are fine, but they are potential vectors of disease for my collection. But I'm thinking I can breed those two together because they both have crypto. Yeah. And we did studies at West Liberty that showed that crypto positive females lay eggs. They don't, like, the spores have the potential to end up on the egg, but we didn't find any evidence of that. And that's a species that's not that, I mean, it's not super rare, but it's not overly common.
Right.
And it's really hard to find adults. So, like, it's a complicated decision-making process. It does not involve kerosene in a match.
Yeah.
Yeah.
So that's an example of what I would do.
In terms of the serpentoviruses, I would say it is very much context dependent, and I can tell a couple of stories of a couple of different viruses that all had very different outcomes in terms of how I dealt with them.
Yeah.
But I have some snakes that I know are positive, like I have a reticulated python and some Kenyan sand boas that are positive for a virus that I'm not scared of at all. I've had these animals for 6-7 years, and the strain that they haven't passed it along to any other animal in the colony, despite being in the colony before the first serpentovirus was even published. So there are plenty of circumstances where positive does not is not a means of not automatic death. But I have another snake that was a wild caught Papuan python, and that one came in as an asymptomatic positive, but then I isolated its virus in culture, and it was a badass virus. Like it knocked out stuff at a level that I had not seen up till that point. So that was a snake that I opted to euthanize because when I see a virus do something in culture that I hadn't seen, that's scary enough. So
and it's awesome that you have the ability to do that.
Yeah, right. But then I have a group of green tree pythons. I've been trying to build a smallish green tree python colony, and man, it is really, really hard to find a negative green tree python unless it's a neonate. And so I have a whole room of virus positive virus positive green tree pythons that, I'm not particularly worried about the virus these guys have because they seem to be fine with it. But I also have a whole other room of negative stuff that I put a lot of work into making negative. So,
Yeah.
That's one where I don't feel the need to euthanize those animals because I have some place to put them. But if I didn't, that's where it gets a little bit more tricky. And that's where, does this need to go out as a pet to someone who literally just wants one pet snake?
Sure.
Does this, need to live in a display cage in my living room? Like there's a 1,500 options of managing it depending on, what level of risk you're willing to take and how much of a risk you view a particular virus as being. So,
Excellent. Trav?
And I mean, I'm basically in the same boat. I have been lucky in that I haven't had any animals pop positive, but I also haven't tested my entire collection. I've, if I have seen something that has made me concerned, I've checked. But I've also gone kind of the way Zac said. First eliminate any other option before you go straight to the worst case scenario. And
right.
The tests that I have run have been more, I've eliminated all of the other possibilities, but at the same time, this doesn't really look like a true fire burning infection. So let's just make sure. And those animals came back negative. So I do consider myself lucky in that regard. I don't have a giant collection, so maybe that's, part of how I have gotten away with it. Maybe part of it's also because of my just insanity with my quarantining techniques is I keep my stuff really, really separated to make sure until I really, really am comfortable. But if I did have something hit hot, it would be a matter of, of a case-by-case basis. Did it hit hot because it was sitting there hacking up mucus and blowing bubbles? And that's when I tested it, and it looks like this is a really ugly thing, and it needs to then be eliminated out of the collection? Or did it have a sniffle because it was under a stress time of year? I was pairing animals up and it started getting a little bit of weirdness. I tested it. It was hot. I would probably isolate that animal. I would see how it develops. I would, if it had been paired with another animal, I would test that animal too.
Yeah.
I don't, I'm not breeding for sales or anything. I usually just am breeding for myself, and what I sell is just the byproduct. If it's animals that I can get away with going some other direction to still get the same result out of animals that I know are clean, I'll just retire them as breeders and keep them as pets isolated.
Yeah.
If I'm still looking for a specific product that I can only get out of those animals, then I will only use animals that I know are carriers, even if they're asymptomatic carriers. Their eggs, once they're laid, that virus isn't going to be able to perpetuate through the entirety of the incubation. I might give a light swab on the outside of the egg with an alcohol wipe or something just to take down any burden that may be there. But after the end of that, those neos are going to come out. They'll be clean, and they'll go into the neo rack, which I know is independent and excluded from those positive parrots. So I will be essentially purging in that regard.
Excellent. That was actually, you guys basically said everything that I personally think. I mean, Justin, do you have anything you might want to add?
No.
Yeah, I mean, the very, very similar thought processes on all of this,
Mm-hmm. Like I said, I think a lot of it's just being smart and paying attention to how many animals you're bringing in, who you're buying them from, just using your brain.
Yeah.
Is there such a thing as disinfecting too much?
Yes.
Like tools and stuff.
Oh, tools? No. But I think if you try to clean your animals too much, you could actually cause problems by having overly clean animals, which makes them more susceptible.
Sure. I just mean absolutely. Like tools and the things you're using on a daily basis in your rooms and things like that.
Yeah, the stuff outside of the enclosure, I don't think that you can sterilize too much. There's this really cool study that the St. Louis Zoo did, and it's not published anywhere. And I wish it was, because it's really interesting. But they did a study where they looked at corticosterone levels in snake. I think it was blood. I don't know if it was poo. I think it was blood. And cortisol is the stress hormone. That's when everybody's like, I'm so stressed out. What you're really saying is my corticosterone levels are peaking. So, anywho, but they studied cort levels in snakes that were allowed to live in rack tubs with like cypress mulch, and the fecal material and everything was removed once every 2 weeks. And then they did it. And they had another group where they were removing the feces, literally, like snake goes to the bathroom, it's removed. And they looked at the cortisol levels in those two groups of snakes. And what they found was that the uber clean group was massively more stressed out with the cort levels than the group that we would look at and basically be like, that's kind of gross. So I'm not saying to let your snakes slither around and crap. But what I am saying is there's something to be said about spending too much time in that enclosure. That's basically what they were trying to get at. And they actually had an objective data set that kind of showed it.
I got you.
So, leaving a little bit of poo in there is probably okay.
Yeah, and Phil and I have talked about that too, just for the sake of, some animals. Like I noticed that with a lot of my stuff, as soon as I do a substrate change, they're dropping something. And then Phil's talked about with his cobras, how they just coat everything.
Yeah.
And it's, we firmly believe that it's some sort of, not necessarily marking their territory, but a familiarity, this is their area. And that's, that's how they associate whatever with the, the tubs and stuff. And I don't know. That's the only thing that makes sense to us, at least. But
yeah.
Our non-scientific minds.
Your minds are more scientific than you think.
Well, I mean, you're
doing a good job, dude.
in the scientific circles, we call this the hygiene hypothesis. And basically, it shows that the more clean you are, the less healthy you are, because you're not exposed to these things that stimulate your immune system. So it's good to stimulate your immune system some. And yeah, being overly clean can create a problem.
You hear that, Phil?
No, what I miss?
Go take a dump in your bed and sleep in it, and you'll not be stressed out.
Fair enough. Is that the classic?
Not going that far.
I had to take, my contact was driving me crazy, so I switched to glasses. Forgive me for missing the wonderful feces talk. Is that the classic example of do turtles have salmonella? Well, ma'am, if you put your child in a kiddie pool of its own feces for 6 weeks, it will have salmonella.
There's that too, yes, but wasn't entirely what I was getting at.
Right.
Alrighty.
I think we covered just about everything. I know it's getting late here. I think Travis is the only one where it's not. Oh no, it is. We're all on the East Coast.
Yeah, we're on the East Coast.
Yep.
Well, I think this is great, guys.
Yeah, I'm glad we made this happen.
No worries, it was fun.
Yeah, happy to come on.
Yeah.
And it is very, I don't want to say reassuring, but it's definitely a peace of mind to know that we're all in this together. We all share similar thoughts. We all share similar worries and paranoia, but there is answers, to be had, and there will be more and more answers in the futures to come. And I think that's all great.
Yeah. I also think it's a good take home is it shouldn't necessarily be a giant stigma that you have a collection that has some kind of pathology in it, because when you get to a certain number of animals, you're just begging this to happen, because this is literally why diseases evolve. Population, it's a part of population regulation for crying out loud.
There are those who have colonies with pathogens in it, and there are those who don't know they have a colony with pathogens in it.
Right.
Steve
Yeah,
nailed it.
there are those two types of people in the industry. Those who know that they have a pathology, and those that are just they have it, they just don't know it yet.
Yeah.
So we shouldn't be. You shouldn't be branded as, oh, they got crypto in their collection. It should, if somebody's willing to say, I got crypto in my collection, but I haven't found it over here, I feel like that should be the standard, not, oh, we found out they had crypto and then they get blasted on social media and, all that kind of jazz.
Yeah.
You just need to kind of get some communication there. Now, granted, if you're selling snakes that have crypto, that's that's different.
Yeah. Yeah.
But the people that have the courage to come out and say it, I don't think that you should be like, oh, I'm never buying anything from them necessarily.
And that was like I was trying to get at earlier. I think there's too much stigma going both ways. I think people that want to test shouldn't be blasted for testing, shouldn't be called paranoid, shouldn't be looked at as trying to bring breeders down by, just buying snakes to find out if they're positive so they can throw names out there. At the same time, I think if somebody isn't testing every single snake, you shouldn't be blasting them either, Test when and where necessary.
Yeah.
You get to decide what when and where necessary is for you, and nobody gets to shit on you for that, no matter what that decision is. Unless, like Zac said, you're knowingly sending out things that are bad,
Right. Steve?
Yeah, I think that relates back to any sort of, buyer-seller conversation and making sure everyone has the same understanding of what exactly the expectation is on the animal they're buying.
Yeah. Yeah, it's good.
Very
points.
good.
All righty.
Gentlemen, thank you for everything.
Thank you.
No worries.
Absolutely. Thanks for having us.
Thank you very much.
Yes, this was episode 102. Snakes and Stogies brought to you by Puget Sound Pythons. Follow them on Facebook, Instagram, YouTube, all that good stuff. And we will be back for THP on Thursday. It's going to be the last episode of the year. It's going to be me and Jake. And I don't know if we're going to have another Snakes and Stogies. I mean, surely we'll have one more Snakes and Stogies in here. But
yeah,
we got to figure that out.
Yeah, we'll discuss.
Cool. All right. Thanks, everybody.
Thank
Yep.
you.
We appreciate it.
Later.